to one and all present here. We are in the 5th day. From tomorrow we will be missing you all. Let us start today's session. I welcome Dr. Nagaraj Nagatihalli, Assistant Director, Basic Research Center and Associate Professor of Surgery, Sylvester Pancreatic Cancer Research Institute, University of Miami, Florida, USA. And I welcome Dr. Chandrasekhar G. Joshi, Coordinator of MSc Biochemistry Program and Coordinator of GIAN Program. And I welcome all our participants and technical team. Have a great day. Thank you.
So good morning, everyone. So the last day, I think we're going to miss you all, as she said, today onwards. I hope you all enjoyed it, and you have to take the test, and hope you all pass. Today, I think yesterday's talk was we screen natural compounds. And then we select one of the natural compound, which is active compound, and then we study, right? And you saw, if you don't know what natural compound, if you don't know, what signaling pathway, what activates and all those, you kind of learn those. Also, once you know which the active compound, you know how to study. Okay, you can easily. Doesn't matter where you go back, and actually you can think of the same way methodologies, they're all same. So you just start with it. Today, say for example, I have a specific target. Okay, I don't know the compound. I don't know the inhibitor. Okay, but I know the target. So what do we do? How do we do the research? How do we find the compound that can target my specific target that I find, right? So that is the story. I take CREB, cyclic AMP response element binding factor. So from day one, I'm talking about day one, day two. I think we talked about a little bit here and there. It's easy because I can tell you exactly the. Say for example, it may not be CREB, it may be AKT, it may be ERK, it may be other signaling pathways, it may be some other factor in some other cell type. It's okay, okay. But you know the target. Now in it's my target is a CREB, okay. Now I want to find a drug or the chemical which actually inhibit the CREB signaling. That is the idea, right? So first of all, you need to know why you select the specific target, right? For example, I selected the CREB based on many factors. I have investigated, I have done a lot of other research, and then I also published, and I know that this is important signaling pathway, right? That's how I selected the CREB. And also I know what activates CREB. Okay, the same way I told you many things about cell-to-cell communication, and you need to know what activates your target, right? The same way, same philosophy, right? So now we understood CREB is important, and we know all the targets CREB actually, can be targeted and inhibited or activated or something. So now we knew that CREB is important. It's an important transcription factor, actually that regulates many genes involved in cell survival, proliferation, metastasis. It's actually CREB in many cancer, just not pancreatic cancer that I showed. It's also expressed. I also showed you cholangiocarcinoma, gastric cancer, esophageal cancer, And many other cancer, Krebs also get activated in tumor when compared to a normal, right? Making it a promising therapeutic target. That's why it's overall, if you can target Krebs with a compound, it can actually study that same thing, same in other diseases also, where Krebs is getting activated. So that is the idea behind it. So then we thought of doing the compound screening, even though at that time when I found the Krebs, there were not many targets. I mean, compounds that actually inhibit Krebs. There are very few, inhibitors available in the market, even now. But then I thought of doing our own screening and find targets, either kinases, or just the natural compound, both actually. We were interested in all, anything that can hit the Krebs, right? We know now Krebs is, one of the therapeutic target, and we understood the role of Krebs in, cancer, just not pancreatic cancer. We studied all those things, we published also. But only thing what we did not publish is that drug therapy. Means we didn't have the drug at that time to inhibit the Krebs. So we can do knockout, we can do, knockdown, we can do siRNA and all those things during that time. I mean, I'm talking about 15 years ago, right? And overactivation of, Krebs also we kind of screened all the TCGA. TCGA actually came out long ago. I think I showed you one of the figure. That database was there for long. So we were able to see the differences of Krebs expression in different cancers also at that time. So, and also we kind of rationalized why we want to target Krebs, right? So we kind of understood the pro-tumorigenic role of Krebs, and also during the after the progression, metastatic. You saw all the data where we overexpress Krebs and actually it metastasized, the cells metastasized faster. But only thing is I did not go through the molecular aspects of those studies because it's again another avenue. But we were interested in the preclinical development and the clinical development. So also I showed you we knew preclinical studies also, like for example, it indicated us that Krebs inhibitor is important, right? Then we started thinking about the compound libraries, right? So, synthetic compounds, I was just started thinking like, synthetic compounds, can we extract from natural product during those days I'm talking about. And also we were looking into FDA approved, drug library. There are libraries available where FDA approved libraries, even in India you can actually buy it. There are libraries available. You can actually screen those libraries. Like it's all in the 94 or the 384 well plates, 364 well plates. And also you have to think about the selection criteria. It's okay, for example, if this FDA approved drug library, if this is a natural compound library, natural product library, if it is a chemical library, how do we do the selection? How do we predict from this screening, these are the compounds are important, right? We had to do the cell permeability assay and pharmacokinetic profiling, all those things. But you already know, yesterday we talked about all these things. Then you have to design assay. For example, how do you screen? First of all, you know the target, but how do you start screening? How do you take all these compounds and like Krebs inhibitors there? How do we do it? So for that, you have to think about like, first of all, you have to understand like, how do you measure the Krebs DNA binding, or you have to use some fluorescent binder, or you can do, cell-based assay, but you can actually do reporter assay. So reporter assay is very easy. At that time, we were thinking about the reporter assay because we can actually, there is a luciferase tagged, Krebs-driven luciferase tag and GFP reporter systems available. So you can actually tag them and it actually gives you a color. And when the drug hits, then your color changes, things like that. So you can actually measure those color, fluorescence, things like that. So we designed the, cell-based reporter assay for the Krebs. And then we kind of optimize those, whatever the luciferase tag does, cells for the sensitivity, specificity, and also robustness, using the known CREB inhibitor. We already knew there were some few inhibitors available, at that time they were available. You had to compare though. So for example, if you don't know any inhibitor for the target, it's also fine. You can actually screen it. But for us, we had it. We had 1 or 2 inhibitors available. We kind of compared that. If you don't know, for example, if you don't have the inhibitor, you can actually knock out the specific the target, and you can actually study it too and see, make sure that actually is working for you. Is either way, okay? You can actually do it. And then we have to set the activity threshold. Okay, if the activity, the CREB activity in the luciferase activity assay, I know how much activity is actually is good enough. This kind of a threshold, you set it up. If you don't know, let me tell you, if you don't know, if you know the chemistry lab who actually knows the activity assays, they regularly they do those, and you can actually talk to them and discuss with them. Okay, that's what we also did. Okay, in the initial stages, and also you have to know the criteria for hit selection. You have to know the IC50 values, efficacy, selectivity of our CREB inhibition, and also initial dose response curves to determine the activity. You have to know all those things. You have to do all those small assays and determine, make sure that your activity or whatever that you want to do, conduct experiment. Because I'm giving you a hint that there is a validation of even before you do the compound screening, and for your target, be ready so that your target is like all the validation are ready, and now you want to, okay, if the validation is ready, I want to go in and do the compound screening. And also there are some secondary assays that you need to think about. Okay, like using the alternative assay formats, like for example biochemical assay, cell-based assay to validate the CREB inhibition. So you can do a lot of these things like groundwork before you screen. Also, you can do the counter screen to assess the specificity against other transcription factors. For example, CREB you know already, so take another one and make sure that is not getting hit by the when you knock out CREB, and it should not affect the AKT. It should not affect some other ERK signaling or ERK factor. So you have to make sure that it is only hitting the CREB. That's what I'm trying to counter screen and make sure. Okay, that is hitting the target that you wanted. Okay, the validation techniques are dose response studies and all those things. Overexpression knockout. I actually discussed that before. Now we examined whether additional kinases, like for example MEK-ERK and AKT, also activate CREB. We knew in the KRAS signaling pathway, so are also involved in inhibiting phosphorylation. So what we did initial screening, we inhibited MEK-ERK and we also inhibited AKT, and we checked whether the CREB is getting inhibited or not. This kind of assays you have to do. Okay, for example, if you know that upstream factors of whatever the target that you're thinking about, so you better inhibit those and see whether they are the real upstreams or not. So it's just a validation, again, making sure your discovery of whatever the target is actually discovery was right. Okay, that means that you understand the biology of it before even discovering something compound and going forward. Because once you know the target, once you know the compound, you get excited and keep doing the experiment. You forget about, you will forget about what exactly, how they're exactly getting activated also. So we also selected in the initial screening 2 pancreatic cancer cell lines. You can select like 2 or 3 multiple based on, if you want to study the resistance and the sensitivity, you can actually select resistant to and the sensitivity to and check all those. It all depends on what type of experimental parameter that you're following and want to do. Okay, we screened at that time, long ago. Okay, we screened like 80 kinase inhibitors. They were available, and also we had like 35 different kinases from the TalkRisk-Screen kinase inhibitor toolbox. We actually available. This company TalkRisk-Screen, actually you have a multiple FDA approved drugs, and also they have natural compounds also. They can they sell with a plate So we selected at that time. Now we don't have to go with these companies and all. We have NCI, because I'm an NCI investigator, so we can actually get it from the NIH, everything. At that time I was not, so I was actually screening, making sure we get all these things long ago. Okay. Then you plan your high throughput screening. What do? You take all these cell lines, like you actually do all these 384 well plates. You put the cells on the plate, and you do the ELISA and you grow them, and then add the drug. And the drug means that here, when you get the drug, like there is a 94 well plate, you can actually take from the multiple pipette, and then you screen. This is again another technology. Believe me, it is. You can go wrong also. You have to know how to pipette, first of all, in this scientific research. I think Joshi was talking about all those small things that you need to know. I'm not covering those things. What I'm trying to tell you is that, so basically you take the drug. These drugs will come in 384 or 96 well plate. You can actually select based on that, and you can screen. And also there are now in our core facilities where they do it for us, okay, now. And means that the machine will do all those things. It's a computerized machine. Actually, they can do the drug screening for us, okay. They can do everything. And then you, because this is a luciferase tagged screening, so basically you can get the target kinases, where some kinases will get activated, some kinases get inhibited. Okay, you have to think what is actually your research is. Are you activating it, or or you're inhibiting it? You can select based on that. All this high or low expression of CREB, and you can select those libraries and start thinking about it. You have to do research again and start searching for literature and see what they do. What we did is we selected one cell line, we screened, and we got these numbers, right? When there is a high number, there is a low number, there are non-selected one, there is one activated ones, these are all blue in the activated one. We actually have a total CREB expression. We compared that with the phosphorylation. It's the same thing you do too in a smaller quantity. We actually did the kinase inhibitors. These are all the kinase inhibitors we selected, and we have these kinases here. So these are inhibitors, and these are kinases. So we can actually inhibit using all these inhibitors, and but your cells, your CREB, luciferase tagged cells are in the plate. So that means that whatever you hit, it is actually you're hitting the CREB. Okay, so either the activity will go high or the low. So you have to measure those. So what we did, we measured the peak. We measure all the peak, and then we select from this. We normalize those, and we select the best out of it. So then we get that inhibitors of this phosphocREB. There are many. Like many compounds will inhibit all these things, and then there are some activators. Some of them will activate also your CREB. Okay, some will inhibit, some will activate. Okay, now it all based on your research, how you think, what you think about this signaling pathway, and how they are actually these signalings are related to each other, and whatever that you think, you have to do the research again. Okay, you get great data, but what do you do with the data? So that's again research. So you have to think about it, and you have to have a plan and discuss with others and those who actually screen like this, and do it. Okay, and then one cell line is over, and we select the second cell line, and we make sure that we get the same hit or a different one, and then we start thinking about why different, why same. These questions will come. What mutation these cells have, and what is the proliferation rate, and how these cells behave, very important, okay, understanding all this. So then we get this hit, then we select, take these kinase inhibitors, and then we hit it, and then we got this peaks, and the same like the other cell line, and we select the inhibitors for the phosphocREB, and also activators for the CREB. We select all those things. Now we combine all of them, okay, and we get new targets. There are many of them, okay. And we know these signaling pathways. These are known for CREB and well known. I can tell you each one and what they do on the CREB. And these are unknown. These are not knowing at that time that what these unknown signaling pathways do on CREB. And these are completely unknown pathway that activate CREB. Okay, because CDK Aurora, there was no literature available at that time on these ones. Okay. So you select based on this. Now, the next questions will come to you is that, okay, what is the difference between these two cells? And we got this common signaling, because what I'm saying is that there are common, pathways that you selected from these two cell lines. Like that, for example, there is a sensitive cell lines and resistant cell line. You also select based on the common, these cell lines. Just one cell line for the sensitive or the resistant is not enough. You have to make more cells, okay, different cell types, and also you screen all of them and select the kinases and the targets, and then think about it. What's going on? Remember, it actually, what you're not seeing is that they are inhibiting CREB. These are signaling pathways are activating CREB or the inhibiting CREB. In this case, they are inhibiting. Means that when you use the SRC inhibitor is also inhibiting CREB. That's what I mean to say. Okay? When you're taking the Aurora inhibitor, the inhibitor of Aurora is inhibiting CREB. Okay? Means that if you don't have CREB inhibitor, you can actually use CDK inhibitor that also should inhibit CREB. So then you go back to lab and start using the CREB CDK inhibitor, which is FDA approved. Those compounds that we screened, all FDA approved compounds, and CDK inhibitor, one of the FDA approved compounds that we used, and now we can use that compound to inhibit our CREB. That means that if you don't have a CREB inhibitor, you can use the CDK inhibitor and do your experiments and conduct your study, but it still is working. But it may not be specific to a CREB, but CDK is also get activated during the progression of the cancer, so you can use it. Okay, CDK also get activated, and EGFR get activated, SRC, TJ beta, ROC, IC, we are all getting activated. We talked about some of these during all these 4 days. There are these activators or targets are keep coming back because they're all common signaling pathways in pancreatic cancer. That's why they keep coming back. Probably if you take different cell type like breast cancer, lung cancer, probably they are different. You actually don't see them. These ones are coming back up again and again, okay? Okay, now the story is that there comes Joshi. So what he did, we had this screening, we were all getting ready, all the screening came. Then he said, oh yeah, I got the ICMR fellowship, I want to come and do, I gave a letter, and he got this fellowship and he wanted to come. So then we had to fix the time during when he should come, and when he should be able to start some of this work. And based on those things, I think when he was here, I think he came all the way to here. I think there was a lot of water, and then it's Miami is beautiful. It's like a mini Goa. In America, Miami is like for you in India, Goa. Miami is for US is Goa. So like that, it's fantastic place and the beautiful cancer center. And he came there, and it's overwhelming. Okay, it's entirely different. And it takes time. I mean, Satish was there, and the doing postdoc there, and it's an entirely different thing. So it's a great opportunity for a faculty, from Indian faculty coming to US and learning. It's not US, any other country. You go to developed countries, there is different approach they give. That different approach they give to science. That's one of the reasons I was also passionate to go to countries like that and to learn. Okay. What he did, okay, he selected one of the compound that's called berberin. Okay, we had this natural compound screening to target CREB, and we had multiple of them, and then he wanted to do the berberin. So, and also he selected that berberin, and then very interesting that why he selected berberin, right? Berberin at that time, there was not much literature available. Okay, this is again 2018, and also there was NCI China collaborative studies in 2017. He came in 2018. In 17, there was big literature, big program was going on between US and China, where they were interested in the Chinese medicine, okay, on cancer research. And they were working on those, and the NIH was interested, and they were like, there is a cancer prevention laboratory, and also at the NCI, they studied the effects of berberin and other compounds extracted from these herbs on colon cancer prevention. That's the only study was there. There was a little small literature the NCA actually conducted the study, and the berberine study was there. And also, he started searching about what is this berberine about, and what does this actually do, and things like that. There was a small amount of literature at that time. And then he started, it took some time for him to adjust and, getting along with other colleagues, and there are a lot of MDs, there are a lot of people, English and everything, a lot of things happens there, and the whole environment is different. Okay, scientific environment, morning to evening, they all talk about science and things like that. So when it comes to that environment, it takes some time to adjust and also study, right? There's a lot of cultural shock and a lot of other things, right, when you go to do postdoc in different country, right? And then he took and started doing simple assays like migration assay, like colony formation assay, viability assay, MTT assay, things like that, just to learn. That's what he's going to do when he comes back and he start doing the same assays, right? So that's how I want him to get trained, so that he can come back and comfortably know how to do. Even though you do the migration assay, believe me, there are a lot of things can go wrong. We can tell you what you can, when I see the picture, when I see the analysis, I can tell you multiple things that you may be right or you may be wrong, okay? Things like that. There are a lot to learn in each assays, right? And then he did all these Western blot. Just getting the right Western blot itself, it takes 6 months. So getting the one good Western blot, it takes 6 months to really learn. For some people, they're technically very strong, right? Some very, very weak. So you have to know where you are strong and where you are weak. But he learned because there were a lot of other experts around him, because there are other postdoc, graduate students, they'll be running every day, and anything problem happens that they can correct him, and they're beautiful Western blots, right? So you see the HPNE, HPNE KRAS, they are normal. HPNE is completely normal. See what happens, berberine did not do anything. And also he started with AKT because it is not just only AKT, he actually did a lot of other things. The one AKT where actually antibody was available, we knew he did a lot of literature survey, and he got something. AKT is one of the probably signaling. That's why he selected AKT, and it did work for him. So you can see in the BXPC3 and the PANC1, and also this is the mouse line. In the KPC that I keep talking about, the KRAS and P53 mouse line, that is the mouse line here. So these cancer lines, in these cancer lines, you can see that the phospho-AKT is going down, but not in the normal line, and in the mouse line also it's going down. So it's beautiful, right? So basically he got the target. He got the target, but the target, what you were thinking about the crab, he did not. See what I'm talking about? Even though he got hit, the crab was down completely, but when he started doing, but he did not get it initially. Remember, we published on the paper that AKT can activate crab. Okay, when you inhibit AKT, you also inhibit crab. I also showed you AKT inhibitor also was inhibiting crab. But somehow this compound, it was inhibiting AKT but not the crab. But we don't know. There are a lot of literature we have to do. But for us, at least we wanted him to complete the postdoc period, understand just not the in vitro assay, we want him to take him to in vivo studies. So what he did, he went and did the xenograft assay. So I don't know, he remembers these tumors and the berberines treated tumors and all. So these xenograft assays, at least he can learn some in vivo assays so that he can come back and do something more on those aspects. So berberine basically it inhibited the tumor, and even though the target that we were thinking about, the berberine, so you can go wrong with the chemical. So don't think that okay, you screened and you got it and it's perfect. No, it is not like that. So you have to validate them. That's what I'm trying to tell you. So when you're validating it, you may not hit the target. Okay, you may hit something else, probably that is related to your crab. The AKT is actually, we published a paper in Cancer Research in 2018. I think he was there at that time. So in that paper we showed that AKT is the upstream of crab, and when you inhibit crab, AKT crab is getting inhibited. So that is this story. So let me tell you, this is I was telling Joshi today morning that last 1 year there were 177 berberine and cancer studies. 177 papers came out. Even in the Nature Communication paper, there's a strong, great papers. But just 1 year, I'm not talking about 18, 19, 20, 21, 22, 23. In each year, probably there are many more papers came out. Just I'm talking about just last 1 year, many papers came out. So when we started, there was only one paper and two papers, maybe a few others, here and there. Okay. When you see something is happening, some of the natural compound and natural medicine, and other people also will be seeing the same thing. Okay. So that means that the winner is who publishes it. Okay. Winner is always who publishes first, right? You have to publish it. Okay. If the data is good, and you should be the winner. No, not others. So you be the winner and start publishing, right? So we did not publish. So I'm just letting you know, because he has to finish the program and came back, and then we did not, because we have other experiments running and we have other targets and other work in the laboratory. So we did not complete. But what his accomplishment, actually he did a lot of other experiments in molecular cancer therapeutics. He published in a co-author and cancer research communication as a co-author, because he was also involved in other projects. So that when he was involved in other projects, so he was also getting the co-authorship. So it actually help him to get other benefit being a postdoc. So right. So that's one of the example I just wanted to give you, that anything can go wrong, anything can go right. When you are screening compound and when you are selecting something can happen, something not, and also your time factor, right? How long are you going to study this? Okay. So if you would have stayed another 6 months, probably you would have published a great molecular cancer therapeutics paper. Probably you would have figured it out, something molecular mechanism. Probably you would have done something genetic mouse model. You would have even in the pioneer in some areas. Probably you would have come back and immediately applied maybe a great grants here. You never know, right? Even though you got other grants, but what I'm saying, the experience will help in a lot of investigators. I'm not saying that you should be in other country to learn these things, okay? You can actually learn the same thing here. There are a lot of great laboratories. There are a lot of pioneers, investigators all around. Look around, there are many of them around. Okay, we communicate with a lot of investigators. They do fantastic work around, but it's just that you have to find the way to reach them. It's not easy. Okay, you can reach outside, but it is difficult to reach here. But you need to find the way to reach them. So how you reach them and how you contact them and how you interact with them is also key factor. There are great laboratories around here. Okay, I don't want to name the institutions. There are a lot of institutions. Even the universities, there are a lot of fantastic laboratories, and pioneers are there. Okay, my pioneers from chemistry is Dr. Rangappa, Dr. Timaya. They were all chemistry pioneers. We learned a lot of things. Dr. Timaya was actually working on he had a 3 project, DBT, DST, and ICMR. All these projects was running in one time. He had a great project. Even the foreigners were coming to his laboratory and working in the laboratory. Okay, and this is in Mysore University. So I'm a biochemist, but I used to go to his laboratory because his lab work was fantastic, because he was actually applying chemistry to biochemistry and biochemical assays, and also biology of it, and tumor and all those things, because he was curious. He's actually thinking internationally, because the people coming from St. Jude's and other universities from Memphis, they were doing experiments in his laboratory, investigators. So I'm just letting you know, it is about you, like finding the investigator where who actually conducting the right experiments and conducting right research, so you can find them. So just giving you an example that it's not about foreign, it's about here also, it's about you. So in vitro validation of this compound. For example, the one example I gave you, berberine, but there are a lot of other compounds that we screen and conducting experiments that I'm just saying X because we did a lot of other different assays, different studies in different compounds. Every day we were thinking about something, right? Well, one will fail, that doesn't mean that we're going to fail. We're going to come back, and there may be other good compounds that maybe they are active, they may be important, and we keep searching. Right? We did get X compound that is actually another compound with a great study, great compound. It's ongoing, and a lot of things are coming out. I just wanted to tell you that it's going to come out. But what I'm saying, we did a lot of in vitro assays. I want to say X so that you know what we did, so that at least experimentally you know what we have conducted. Right? We did the dose response studies. We did the evidence-based studies and transcription regulator. Same like what I was explaining to you, the reporter gene assay And also we did the binding assay, inhibition, Krebs phosphorylation, all these assays we did in vitro. Okay? We did again mechanism of action. So we went ahead, like how I showed you the urolithin compound. The same way we went ahead and did a lot of mechanism of action, just not Krebs, right? How this is the whole thing is happening. You have to study. It's just not, you just take the compound. Okay, fine, it's about your facility. But you need to find a way to understand the whole mechanism, right? As a researcher, scientist, right? And we went ahead and we took this compound and we studied all the cofactors for the Krebs. We did more than we think of, right? And also depends on the person who actually conducting experiment. If they get excited, believe me, the science will change. If they are not excited, then it doesn't matter how much I'm going to tell. It's just not going to work out, right? So you have to get excited. Then only the science will change. Okay? Doesn't matter how much the PI is going to talk to you and tell you. So and also we did a lot of signaling pathway, and also we did a lot of Western blot, phosphorylation, Krebs, different cell types. So we were talking about sensitive and resistant. We also did some studies, sensitive and resistant. Also we did some metastatic study, invasive front, and all those things. And we did chip assay to see what is binding to Krebs and how the Krebs is regulated by other factors, and what is this compound is doing, where this compound is going and binding to activate Krebs, and how this is all the interaction, things like that. Okay, just to give you a hint that what all we have done, so that you also know that as an example, you all can do the same thing for your compound and active compound. So what we do in our laboratory, preclinical development to the clinical, I showed you a lot of examples. When it comes to cancer field, why you want to study anything in cancer? Because you want to cure, right? End of the day, you want to cure. Okay, if you want to cure, then you have to do the clinical studies and preclinical and clinical development. That is the idea of cancer biology. That is the idea of cancer research. You have to understand how the cancer grow and progression of the cancer, metastasis. These are all great. This is all biology. That's also very important to that's what we do. That we understand the biology, and then we go ahead and do the clinical development. And if you don't understand the biology, it is very difficult to study, right? If I don't know what Krebs is doing during the progression of the cancer, we don't know where we are targeting, when we should treat this patient, and when we should treat these mice. These kind of things, you have to have the knowledge of the progression and also knowledge of chemical compounds and the screening and understanding how to study the biology of this. And we also did the pharmacokinetics analysis. So what I'm giving you is a step. When you find something, what you need to do, okay? So we already discussed yesterday, other way, the reversal way. But here, we know the target and we are doing the screening and selecting the compound and studying. So we do the same way, toxicity study, pharmacokinetics profiling, and then we evaluate in vivo, like PDXs and a genetic model. So we have the patient PDXs, and we inject in the mice, and then we study, again use the compound and see whether it works. Translational, like making sure the same thing I'm repeating again from yesterday. The reason I'm telling you, because that study is different, this study is different little bit. If the way that your laboratory or you are thinking, you can actually plan accordingly. Okay, and we finalize a lot of other things, just not biology. Same time parallelly, we select the lead compound and move on, right? So the impact of this next step, you already know. You can actually tell me now what I'm going to do. You know already what I'm going to do next. I'm going to do the chemotherapy, I'm going to do the immunotherapy, I'm going to study further on the tumor microenvironment. You already know. You know my thinking, right? Exactly, it's the same thing. So it's a standard protocol. It's just a standard procedure, okay? Once you know how this work, believe me, you can do plenty of work. It's the same procedure, same thing. Okay, I'm going to keep repeating the same and again and again. It's the same thing, okay? I'm going to do the monotherapy and also do the combination therapy. If you want to do the combination therapy, you need to know what are the activators. You saw a lot of activation, right? There may be activation. What I did not mention there, that there are some activators are very important. If they get activated, it is bad for tumor. Okay, you're using some inhibitor and something is activating, even though it is targeting CREB, is bad. So you need to select that inhibitor, block it with the combination therapy. Okay, block it with the combination therapy, and also you do the chemotherapy on the other therapies, and you do the, now next you're going to do genetic mouse model, and you're going to do PDXs, and you're going to do a lot of things, and before you translate your study to the clinic, right? So you all know now what to do. I hope you're going to start working and start thinking it's a standard protocol procedure. Okay, you just do this, we just do the same way. Probably until unless some postdoc and grad students come smarter than us, and even more smarter, and they're all smarter than us, definitely. Okay, each one has a smarter idea and different ideas. Okay, and if someone comes like that and think about something differently than what we are doing, we will definitely change the way that we are thinking experimentally, right? Sorry, this is not egg, this is a take home message. This is now, overall, like, it's a compound screening optimization, and can lead to a discovery of, effective targeted therapies. I showed you one of the discoveries, uroleuthine, and there may be other discoveries coming. But what I'm saying is that you screen and start, and you discover something that is actually useful to the patient. And also, it actually gives you a hope for improved outcome. Either if it is a CREB-driven cancer or AKT-driven cancer, whatever is driven cancer, right? We are saying that CREB is activated in the tumor, but it can be something else, okay? So it actually gives you a hope. Oh yeah, I know what, I think I can treat this patient even today or tomorrow, someday in the future. I think my compound, my study will go to clinic and cure someone. Okay, I'm so happy for that. I think that it will happen because I'm actually towards that, right? I think that is the take-home message. I want you to have it. I hope you have taken all this knowledge all these days. Something will be definitely, here and there, something will be helpful to you. And take this message, take this knowledge, and go back and I think start thinking what you can do to cure cancer, right? I think that's important. So I'll take any questions if you have, few minutes. Okay, so now these questions you can actually have questions from even the previous days is also fine. So if you have any doubt on a compound screening, I'm not specifically to the scientific, I want to come up with a general. Like for example, you have something that you don't know what to do. It's okay. So you want to start. Where do I start? Okay, it's okay. And you already started, you got stuck. It's also okay. We can actually try to help you, or maybe give you a hint, or we all try to solve each and others' problems, right? So if you have any questions like that, please let me know. A few minutes. Okay, we can discuss later. So thank you so much. I think we have this discussion forum after your test. So that is the final session of today. So in the discussion, we're going to discuss, like, if you have any questions. If not, it's okay, fine. We'll discuss generally. Okay, we can talk about our life. It's also fine. Okay, so we will have that session. I think it's probably the tea time. I just don't know whether they are ready. So we can do that too. Yeah, sure.
As you use this array of kinase inhibitors, so there were 80 kinase inhibitors for covering about 35 kinases. So among these, I am not seeing the CREB inhibitor in that.
Because there are no CREB inhibitor, which. So your question was is that there are no CREB inhibitors in the screening. All the upstream regulators of CREB. You're right. That is our plan, because we don't have the Krebs inhibitor. That's why we went ahead and screened. So if you have the inhibitor already, and why do you want to search for another inhibitor? Rather you study something differently. Or if you want to find a new inhibitor, if you have a Krebs inhibitor like already approved, also you can add it to the screen. You can actually, these companies can add it to the plate and give it to you. So our idea, because we don't have we want to find something which actually we can use a clinically approved drug. That's the reason that we screened all the upstream targets and regulators. So we did find a lot of them, but it's just that, remember, as I said, it is not easy to find the one actually is working for you. So again, you have to screen each one. But remember, there is always something come out from the study that you're going to do, right? Or when you start, experimenting these compounds, I think you'll find something new. But important, I think, how you find, and it's about your experience, and also the laboratory people who actually, whether searching literature, understanding the concept, and designing the experiments better, and the literature survey, and all these things, all what I was talking about yesterday. It's all about that. It's all about knowledge that you have. Then only your science will change, right? You can do the way, for example, my mentor says, it's okay. But there are I was not. I was like, I was always searching for literature, thinking about what is the new idea, new concept, new design, new thinking. So that we were able to discover more and more. So I think important is that giving the next generation, scientists, like for example, grad students or the postdoc, I hope, and also at the same time the awareness and literature survey every other day, and giving them the time to search literature and collection of literature and discussing with them, having a journal club and going through somebody else's paper, published paper, and trying to understand, discussing in the laboratory how exactly they conducted these experiments, so that you learn from those experiments and discuss yourself. It's okay, whatever the knowledge that in that room, it's also okay, you can actually discover something. These are all some of the criteria that you can change the way that you conduct those screening and experiments. For example, we take one of the scientists called Channing Durr. He's from North Carolina. So he is my one of the pioneer researcher that I actually study his papers a lot, because he published a fantastic paper in the field, Nature Science, and also fantastic, even the small paper, it's purely scientific papers. And I learned a lot from the scientists. So even I met him and I talked to him in many conferences we go to. He's one of the pioneer and in our pancreas field, and also in the drug study. He has done a great job in scientific field. So talking to them and learning from them, and also even though he actually went through one of my study when I asked him, okay, can I show you what we did and conducted, and he was so happy to see and give his hope and whatever that we are doing, right? And learning from kind of such scientists is also something that gives you energy. So go to conferences and meet some scientists like that in an Indian scenario, or someone who comes from outside who has experience, and talking to them and taking their time and few minutes and discussing your research and science is also important, right? They all want next generation to be a better scientist than them, right? Or I want next generation to be better scientists than us or me. So happy to help and give whatever the suggestion we can give scientifically, and so that your science will be much better than mine, and you discover more and find better cure than what we have done, and the things like that. So be open-minded, and also same time increase your knowledge and awareness, right? Okay. Let's have tea and then we'll come back. Thanks. Okay. ಟೀ ಬಂದಿದೆಯಾ? years you have to go. Yeah, you can try it. No, it's not that only you have to apply there. There are a lot of European countries also use a lot of scholarships, right? So when you go, then you go to, for example, okay, you go to Florida. Okay, when you go to Florida, there are many universities. Okay, say for example, FIU. Okay, this is on University of Florida International University. It's a small university. It is not like University of Miami where I am from. But here they take a lot of international students. But you need to know about in each state you go to, you have to see how many universities they have and see what international programs they have. For example, you go here, you see a lot of international students. You go to Florida International University, you go to their PhD program, okay, admissions and PhD, something like that. You go each website. Each website they have each website have a search option here. Okay, then every website will have search option. Okay, you go to PhD in whatever that you want. Okay, PhD biology. Okay, then when you go to PhD biology, go to the PhD program. Okay, then every there's a admission process. You can go to admission process. There's a graduate manual. You can go to manual and see whether international students they take or not. And when you go to, there's a deadline. You can actually go here and see December 1 for the following fall. And there's a requirement. And you have to see here for the foreigners, foreign students, what happens. And they also talk about funding. They from the outside, the inside funding, things like that. Just to give you an idea. In each university you have to go in such a way. But don't go for the private universities. It's very difficult to fund it. And also it's very expensive. You don't have to, you just have to do, these universities are much better than IAC. Means that, standard wise, facility wise, I'm talking about. So I'm talking even the small universities, they are certainly universities.
ಅದು ನಾನು ಗೂಗಲ್ ಮಾಡಿದ್ರೆ 1000 ಆಗುತ್ತೆ. ಟ್ಯಾಕ್ಸಿ ಮಾಡಿದ್ರೆ ನನಗೆ ಗೂಗಲಲ್ಲಿ ಯಾವ ಮಾಡೋದು ಅಲ್ಲಿ ಹೋಗ್ಬಿಟ್ಟು ಟ್ಯಾಕ್ಸಿ ಮಾಡಿ ಸರ್ ನೀವು 1000 ಕೊಡ್ಬೋದು. ಇದೆ ಕಡೆ ಕೊಡಲ್ಲ OCI ಕಾರ್ಡ್ OCI ಅಂತ ಹೇಳಿದ್ರೆ ಇಂಡಿಯನ್
Good morning. So, actually, this was a long sessions which were going on since 18th of this month, and we are coming to the closing ceremony of this GIAN program. And as a part of this, the program, we are supposed to conduct the test that is actually maybe the as it is mentioned in the GIAN program, we are supposed to conduct. And in this test, we have framed the questions based on the sessions that were conducted since last 5 days. And you go through the questions and you try to understand, and you have already studied and you have been trained to think about the various aspects of the in vitro and in vivo models. So go through the questions, and before that one, make sure that you have written your name, as well as you have written your, you have put your signature on the question paper, and you have to mention the date of the examination. And this is a multiple choice questions. So you have to mark only one answer for one questions and go through it, and you have to fill accordingly. So we will start the test. Now it is 10 minutes to 12. We will start the examination. So once the test is completed, that is nearly for 30 minutes, then we will move on to the next session. Nagraj. And after that, once the discussion session is over, we will be moving towards the valedictory function. Thank you.
Hello everyone. So this is the last session of this the whole program. This is going to be we plan to have the interactive session. So just to go through overall from the day 1 to day 5 from morning session that what we discussed. So this is going to be more of interactive, just to go over what we learned all these days, and also kind of all the models that we developed, and just to have the interactive session after I go through some of these models that we discussed all these days. Okay, so overall in the model that we discussed about the in vitro models, the cell line model, 3D culture, some, and organoids. I just touched on the organoids. When if you want to do the organoid culture, how you do it, and whether you want to do it, and if you want to do it, like how to do it, there's a procedure. So we can actually, there are a lot of beautiful papers published on methodologies. So you can just say organoids and methods. There are a lot of beautiful papers. So in our laboratory also we follow those methodologies from the pioneer who worked in pancreatic cancer, and then we follow their paper, methodological paper. And the same way, if you have a different cancer, different method, you can actually follow the same. And more over that, there are a lot of discussion forums online that you can actually, if you have any doubt and questions all over the world, people discuss about a lot of problems and issues. So that's what I think during when we were learning. So we discuss through the forums, but nowadays we meet them in the conferences, and we go to the posters and presentations. Organizers who are actually working on, they're all pioneers, so they might have learned a lot. So we go and discuss during the poster session and also during the presentations. So what I suggest, you go to these conferences if you are interested in some particular aspects of your studies, and go to those posters where they've used it already. Search for those keywords, or go through those posters and talk to them and discuss with them. And they are the one actually real biologists like working on those technologies. So in the 3D culture also, it may feel like simple, but it may not be simple. Okay, you just there are a lot of errors and things that when we start working. So go to those posters and go to conference. That's why these meetings, for. So to meet other investigators who are actually working in that particular field and interact with them, right? So in vivo models the same way. We talked about xenografts, we talked about syngenic models, GEMS, patient-derived xenografts. Any of these questions or anything issues, or if anyone wants to work on, there are a lot of these conferences you should go to, symposias, or someone is presenting something on related to somewhere else. See whether you can have a Zoom call or online web media or related discussions. There are a lot of universities. They do have a open format. Like anybody from anywhere in the world, they actually access those webinars, and you can actually listen to them, what they are doing and conducting experiments. So don't have the fear. So you don't to click those Zoom links and webinars. So you can actually, anybody can go to those webinars, attend and learn. Okay, if you are interested, there are a lot of them. You need to start searching. Searching itself is a huge thing. Like you learn a lot. For example, like if you are interested in cancer biology, there is ASCR, right, American Association for Cancer Research. And the same way here in Indian Association for Cancer Research, they may be conducting the webinars, and ASCR conducts a lot of webinars, and also they conduct, for example, a Cancer Discovery Journal, right? They conduct the webinar. Anybody can access those webinars. Okay, means that journal club. Someone from that paper that who published, actually they talk about their paper and technology and how why did they do those experiments. Exactly like how I was actually telling you about my story and my papers. The same way they talk about those papers, and you need to listen to them, understand what they're doing, and if they are, if you're interested in those studies. We did not touch much about bioinformatics, but bioinformatics is a tool. I did talk about R program, other programs, and programming, and single cell sequencing, RNA sequencing, data analysis, and when you run the flow and flow analysis and all this analysis itself, I think you need experts. Okay, so that, and then in vitro model we touched even today morning about the 2D culture, high throughput screening, and a 3D culture and organoids. We did talk about a little bit here and there, and the reproducibility issues and the lack of microenvironment. We talked about all these contents, like why microenvironment is important, when we should use these microenvironment related studies, when we should not. Okay, just stick to a xenograft model and keep going. The important is what challenges have you encountered within when you are conducting in vitro experiments and models, and how have you been overcome them. That's the question that you have to think. If you have not yet overcome the problems, then start thinking about whom to contact and get the information, right, or discuss with them. When we come to in vivo model, in vivo model, we talk about in vivo model because we want to bridge the lab to clinic. That's why we want in vivo model. Otherwise, we can do a lot of in vitro and biology studies, right? So we want to take that to the clinic. So that's why we want to bridge that gap between clinic to the lab, right? That's why we make the in vivo models. So we make different in vivo models, like for example, xenograft models, for a reason, just for the tumor growth, drug testing. That's all. That's all. So if you are thinking about taking a xenograft and looking into some other marker, some other microenvironment, may not be the right model, right? So that's why you need to know what model to use. Okay, syngenic models. So these are immunocompetent system, ideal for the immunotherapy studies, right? So if you want to do the immunotherapy, these are the right models. The GEMS and PDXs reflect the human tumor heterogeneity. So that's why they go for the GEMS and the PDXs, stromal interactions. So these are all microenvironment if you want to study. Those are the models that you want to, do. I shared my experience with the in vivo model selection and all the limitations that I went through during our work and in our laboratory. I've already discussed a lot of things that If you got the information, it's fine. If not, it's fine. So at least you know when the problem comes to you, then what you need to do, right? Reach out to someone who is expert in that particular area, particular model, and things like that, right? Okay, and then the competition biology, very important. Nowadays especially, you cannot just do alone. Science is going beyond. Every other day there's a beautiful papers coming out in publications. So it's okay. If you are sleeping more, then believe me, that sleeping hours, the more like 2 hours extra, the one when someone is publishing another paper. So think about it. And if you want to do research, there is a limitation. So how much you can enjoy, and also how much you have to spend time for each thing, and where you're wasting time, and where you can put those time in what for, things like that. You have to really judge yourself and be competitive. That's all I can. That's what I'm trying to tell you. So when it comes to bioinformatics, genomics, transcriptomics, proteomics, and you need to know those. If you want to do all those things, and you never know. Probably in the future, in another few years, probably you'll be one of the expert in transcriptomic studies or proteomics. Probably you'll be in industry and doing something like all this high throughput omics technology. You never know. So think logically. The models that you use, it doesn't matter what bioinformatics tool you're using it, but there's a logic behind each technology. I've already discussed, like you don't have to do genomics, proteomics, and transcriptomics. It all depends on the model that or the study that you're trying to do. There are a lot of virtual screening, a machine model, predicting therapeutic response. There are a lot of models available. It's about, you don't have to think all of them, study all of them. Remember I told you? So you don't have to. You can learn on the way, but you just focus on something, take some problem, starts working, right? Important is when it comes to a computational tool, to understand what computation tool have been most impactful in your research. You need to find that, and also you look for someone expert in that particular aspects of computation tools that to use. For example, R. Why you want to use R? Okay, what for? If you do this analysis, what computation tool you need to use, and who is your friend in the bioinformatics department or somewhere else in the bioinformatics, or the computers, someone like in statistical analysis, someone who knows actually statistical tools, things like that. So it's all about collaboration and thinking about friends and colleagues and around the world or around India. So therapeutic strategies, we talked about small molecule inhibitors, monoclonal antibody. We did not touch in detail monoclonal antibodies. So all you can talk about that if anybody is interested. It's a gene therapy. We can talk about targeted when it comes to targeted therapies. Immunotherapies, we talked about immunotherapy, checkpoint inhibitors like PD-1, anti-PD-1. There are other CAR T cell therapy, cancer vaccines, and there are a lot. You can, if you think about, if you want to do the CAR T cell or the cancer vaccine, you can learn vaccine, how to do the vaccine, and go to that field and learn about it. It's all about your interest, though, end of the day. When it comes to chemotherapy, radiotherapy, these are all conventional therapies. They are still in use. They do in the hospital. That's all they do. Chemotherapy, radiotherapy, or surgery, and then chemotherapy, radiotherapy, adjuvant therapy and neoadjuvant therapy. They call in their terms in the clinic. So if anybody patients in the hospital, you can hear all those adjuvant therapy, neoadjuvant therapy, and these kind of things. You as a cancer biologist, you need to know all those words. Important. The clinical words are important to understand what exactly is going on in the hospital, so that you have to mimic all those things in the laboratory. And when it comes to combination therapy, right, integrating multiple modalities, and also I talked about overcoming resistance. I started the talk with the overcoming resistance. The reason is, I'll tell you, I told you that the pancreatic cancer, right? So the same way that different cancer have different reasons to start with, and also when it comes to multiple modalities, integrating different technologies, studies, and models, and we talked about and touched all those things. So also you need to know that which therapeutic strategy, has shown the most promise in your study is a key thing, right? So you need to know what strategy you should use in the therapeutic strategy you should use it during in your study. So like you need to find out your own way, and also the key aspects, concepts are in the targeted therapy, focusing on the molecular target. So for example, we talked about one particular kinase, one particular transcription factor, oncogene. So think about it. I talked about KRAS, talked about P53. Think about why you want to target this. I gave you a lot of logics behind why I want to use KRAS model, why I want to use a P53. The logics behind. So you cannot just go ahead and start working on something like I want to start. There's a logic behind in each and everything. That's why this takes, you need to do a lot of literature survey. That is the reason. So, for example, we talk, there's a EGFR inhibitor. In our screening, the EGFR came up. PARP inhibitor people use, BRAF, inhibitors they use. So these are all targets. So you need to know what target you want to touch and why and all those things. So there are. You also should be able to judge yourself how you want to address this resistance pathway. So in any cancer you take, resistance mechanism is an important thing when using the targeted therapies, right? So you need to know when this tumor will be resistant to some therapy that you are doing, and how do you overcome. So I gave you a lot of examples in overcoming resistance in our models, and you think about your models and your studies in the future. Okay. The types of immunotherapy, there are many types of immunotherapy. We use one, I presented one anti-PD-1. There's a paper that we published, Cancer Gastroenterology. We used even CTLA4 in that. There is another checkpoint inhibitor. There are cancer vaccines, cytokine therapies. So we have done cytokine therapies too. So there is a reason why we do the cytokine therapy, and also which cytokine we use as a therapy. There are benefits, limitations in each one. If you come to immunotherapy doesn't work in the pancreatic cancer, just the immunotherapy. You have to add with the targeted therapy, chemotherapy, and other therapies. So there is a reason we can, like as you do research and establish papers and knowledge, I think you will know why I'm talking about all these things. Also, have you planned to integrate the immunotherapy approach in your research? If you are like I've given you example where my work actually took us to immunotherapy. It's not we wanted to do the immunotherapy. During that time that we didn't know what's going on, then immunotherapy came, and then our survival was better. So like that, there is your successes and challenges. There is always comes in between. There is a success, there is a challenge. Okay, you are successful, and then you be happy, and then the challenge comes. So it's very common in research. So when it comes to combination strategies, overcoming the compensatory mechanism. So it's very important. When you target one, there is another signaling pathway will get activated. It's because the cancer cells want to survive. That's why the other signaling pathways get activated, and they want to survive. It's very important, very common. So that's why you need to understand the multi-signaling, which pathway is getting activated. Also understand the heterogeneity, like I was showing you. I tried to target the tumor intrinsic pathways, and then we did not get the survival. Then I went into heterogeneity study, and then we started inhibiting heterogeneity, whatever that immunotherapy, and then try to do the multiple approach to hit the cancer. So combining targeted therapy with immunotherapy, I gave you examples, things like that. But important is that how do you design your combination strategies, why you want to design combination therapy. Sometimes your data takes you there, sometimes you design and then see what happens, things like that. And also you need to think about biomarker study, that we did not touch much about it in the patient care, and biomarker study after you do the treatment, and to stratify the patient. Basically, you do the biomarker study after you do the therapeutic studies. You collect all these blood samples. There are different ways. There are many ways that you can do biomarker studies. Translation research very important when it comes to a cancer research. Yeah, that is the bench to bedside we always call. So and then sometimes it is important to also take from the bedside to bedside. Means that the patient samples, patient blood samples, patient data you collect and come back to the lab and analyze them and see what's going on. And then you start thinking about the science. So that you can actually say bench to bedside, bedside to bedside. So it's important. Both sides are important. So think both sides. If you have opportunity to work with a clinician or the doctor or someone, it's actually great because you're actually talking about the bedside to bedside. You as a researcher, you bring those samples back to the bedside and then start thinking about your research as a clinical translational. So integrating the preclinical model to therapeutic strategies. I talked about today morning, like why you want to do the in vivo studies, because you want to take to the clinic. That's the reason that you want to do the clinic in vivo studies. The same way you have to understand the preclinical models and also strategy, therapeutic strategy to. So that will tell you what's going to happen in the clinical trial. Maybe it will work, maybe it will not, but at least the logics that scientific logics has to be perfect. So the importance of biomarker development, patient selection, and adaptive trial design. These are all going forward after phase 1 trial, and further when you go to the higher up, phase 2, phase 3. These are all comes in. Don't worry right now, but I think important to know all those phases. And there are challenges when translating your laboratory findings to the clinical setting. There is no doubt about it. Okay, there are a lot of learnings and there are a lot of interaction you have to do with the clinicians, the doctors. So don't leave anyone. So any hospital, I was just keep telling, there are many hospitals here. There are many clinicians. They are knowledgeable, much more than what we think. Okay, so we are just a biologist. Okay, the information out there, what they are seeing the patient every day, and they have a huge information, real information. Okay, but what you have is the brain of thinking about biology. So you need to collaborate with someone like that, and then challenge your research, what current research you are doing, and challenge yourself every time. That's most pressing, important thing. How do you, envision the overcoming the gap between the preclinical models and the clinical outcome? You have to start thinking yourself that these models. If you want to model this also, you need to have the collaboration with the clinicians or someone who actually knows. For example, I wanted to design the chemical library, and I went and had the discussion with the INSTEM, my friend. So INSTEM, he was able to help me. So because like that, there are many of them. Okay, like different field. Pharmacology, there is another person called Raj. He's in Minnesota in another university, and whenever I have an issue with the pharmacology, I talk to them. So it's important to know pharmacological side and how to develop the drug and the problems and issues. And same way in food technology, you talk about the packaging I was talking about yesterday, and also there are a lot of other things that issues comes. And I know those process also. As when I was in CFTR, I saw that the whole. In the spice and flavor technology department. So what I'm saying, each component, each when you want to design, there are always problems. You have to overcome those gaps and problems. In also cancer research, there is from preclinical to the clinical outcome. There is always a gap in between problems. So we think about all those things, and when you are designing your experiments. So again, our last take-home message is there is no single model. Therapy is universally effective. Okay, so do you think that this model is perfect? There is no way. So there is another model comes out, and other model may be much better than you think. There is nothing called like single model is important. This is effective. If someone talk about one model, this is a great model, blah blah. It's okay, fine. You accept it and you move on and you do your best, okay? This is what we did, okay? We did whatever we think that is best, okay? Integrated approaches tailored to the specific cancer type and patient profile are essential, because you have to integrate approaches. Like means that, so in each cancer specific, there is an approach you have to give, okay? In my cancer, I understand how I have to do, okay? The same way, if you ask me, prostate cancer or some other cancer, so I just don't know how to do it because I'm not the experienced investigator. So, but I only give you a hint, whatever I know from my cancer, give you a hint. But probably the specific cancer type, understanding it, and also the specific cancer patient profile, understanding that is, I think, a key factor, essential, okay? Whatever I said and talked, okay, whether you're using it or not, but you will use it in the future. You have to start thinking today, so that it takes years to establish those concept ideas. It is not today. I know that you will not be able to today, but do it today. But the ideas and the concept and designs, how we think. How we think about the problems, and what and all you need to think about when you take one problem, right? One cancer, the same way, one problem, your scientific problem, it's those logics, I think, key thing overall, right? That's all. So we are done almost. Okay. Thank you so much. Now, a few minutes, we're going to have, interaction, few questions, final questions. After that, we'll disperse, and if anything, we can discuss later. Okay. If anybody have any general questions or anything about, anything, it's fine. Okay. Yeah.
So it's not about science now. I think you told everything.
Yeah, it's fine.
Tell us about your journey from a small village like Nagatihalli to the US now. How is it? How was that? And what are the guidelines you want to give to someone who is aiming that?
So I'm a village boy. Okay, I studied up to, I think, 7th grade in my village school. My school has 100 years of celebration next week. Also, I'm doing that. I'm going to my village and we'll be celebrating for 3-day or 4-day function in our village, and there'll be a lot of students come, and almost everyone probably trying to be there. And that is a huge thing. And after 7th grade, I think I was not a sport boy. I was always kind of when I now if I look back, and it's like a village. Anybody from village is the same thing I have experienced, right? I have to take care of the home, and we had a goat and bullocks and all those things. Same thing. We had to take care of them. Saturday, Sunday, we had to take them to coconut trees around, and we had to clean them. We had to put water to coconut trees. It's not easy. And also we had to take all the clean everything, all this goat and everything. We had to take those to the trees. We had to walk all the way to 2 kilometer, 3 kilometer, and put them back and come back and have a breakfast and go to school. And then afternoon, same thing. It's the same story, right? And we do all those things. And then I went to high school. In the high school, I think I went to Navodaya in Chandrapatna. So I was supposed to be in the English medium. They put me, there is an entrance exam for that, and I was supposed to be in English medium. So according to my dad was a teacher and headmaster, so he put me in, at least you should study. So, but I went into English medium, and then I realized that I was not able to, get anything what they are talking about. What I studied, social studies, is different than what they are talking about. I have no idea what those words are. Then I passed and I went to English medium, and then it came back. And I went to headmaster, and his name is Puttaya. I still remember I did a namaskar, and then I told him that I will not be able to really understand any of those words, and I want to go back to Kannada medium and study. And then he said, okay, you know what, he was so happy that someone is coming and wanted to go to Kannada medium, because everybody wants to study in English medium during that time. And then I went back to Kannada medium, and he was so happy, and then put me in one section. And then I studied 3 years. My parents didn't know that I studied in Kannada medium until I got the 10th marks card. And I was a union leader, and I was a president of the union during that time. So my dad thought, I think he's going to go, I think become something politician or something. So then he put me in Mysore, some hostel. And then then he was really worried that this guy is going to go in other direction. So there was one thing that I disciplined myself, because I joined NCC during my high school. I was really a kind of a social boy, and NCC actually helped me a lot. I came to Mangalore during that time, integration camp, when I was in, I think, 9th grade. So in the Canara school here, I think I had a national integration camp. I think that changed my mind. That means that, when they call me Mangalore University, now you know why I'm here, because that inspiration, that when I was a small kid, when I had that integration camp from Punjab, from Assam, there are many students came from all over. I really saw all these people, different, language, Hindi, and many things. That was actually, I never went out anywhere other than like Chandrapatna and then Bellur, and that's all. That's it. That's my whole, understanding of the whole world. And that, I think, integration camp helped us. So you all here from Kerala and other places, and I think that is actually helping you, because you're seeing lot of things that you learn, right? So young age, if you get to know all this integration, of different culture and things, I think that changes your mind, right? Probably my mind was changing. I was actually leadership, little bit qualities that during that time. Also, I was doing the Hindi, Prathama, Madhyama, Pravesh. I was learning English, Hindi during that time, so that helps me. I was always kind of because I'm in NCC, I was maybe thinking I want to join army if I don't get any job. Kind of that culture, that the high school is like, blood, how that flowing, like, you want to, do everything. Then when I went to Mysore, right, that is one of I never left Mysore until I did my PhD, even though I got a lot of opportunities outside, when IRA and other things, but I came back always Mysore because very attractive education-wise. I think that place for me at that time, there were a lot of things that I learned. I used to attend a lot of lectures from different places, and also different colleges I used to go if their lectures or anybody is giving a talk or anything. It's my own discovery. You know what I'm saying? So when you're in a hostel, you have a bicycle, and then you just keep going anywhere you want, right? This hostel was like only 2 meals, morning and then evening. That's all. There is no in between any meal. My dad used to give me $225 rupees. Then 25 rupees extra, that is for the bicycle, 250 rupees actually every month. During my PUC, my English medium. So basically, I studied, I told you, I think with a dictionary, I was always keeping my dictionary right next to my, physics book and these things. I never went to any coaching classes or English, any mathematics or science, even though I tried to go, but there was no money, you know what I'm saying. So all my sister and brother, everyone was studying, my teacher kids, and I think it's very difficult for everyone. Everyone was studying. My sister was studying in Mysore, and my brother was studying in Bangalore. Things like that. It's very difficult for a teacher to, afford everyone. So we knew all those things. So kind of it's difficult for the tuition. So I learned myself everything because I think that hunger, I think, makes you better, right? It's important. Even though we say that, when you are giving everything to our kids, what happens is probably without hungry, I think it's very difficult for them to understand the value of the food. So after the PU, I still went to NCC because that is my passion. probably at least I will have a job in army, something like that, if I don't get anything. So went to, after PU, I did not get that great score, so I had to go to BSc during that time. So at least I thought after BSc I will do the medical. I was very much interested in medical school. I was so passionate about medical school. And I didn't know how to do the CET exam. Believe me, I didn't know the training, because I didn't have money to go to CET classes, course, or anything like that. So it was difficult. So we didn't get the score. So I was, I was not the great student probably at that time. But I was focused. Whatever I learn, I always get it inside. That's what I think. I attended classes, I think, regularly. I did not miss classes. So any classes, whatever I can get, 1 minute, anything, the knowledge I get from the classroom, I think that actually helped me a lot. One of the English teacher, I think, I studied in JSS College in Mysore, PU, and then went to Urajas College in BSc. In the PU, I think one of the English teacher, I think, attracted me a lot. He was actually teaching me English in Kannada style. Kannada to English, English to Kannada. So that actually helped me a lot. I had to really salute to him, because that was needed for me at that time. Because that I was actually transitioning from my own language to English. So that's, I think, the key important thing. And there was a mathematics teacher, one of the lady that actually attracted or the way of teaching and all those things. So those kind of small things helps you all. I think you have the same type of experience for sure. You have a favorite teachers. And during my BSc, I think one of the research that I want to take today, because of that my teachers during my BSc class, BSc time, there was a Panduranga, his name is Dr. Panduranga, Dr. Munyappa. He was a zoology teacher, okay. So Dr. Panduranga used to teach by taking the science and nature published papers on the front page, okay. When he was teaching me nurture and nature, he used to talk about, okay, how you kindle in the classroom. He used to take about nature, nurture, and say that this is not from genetics, this is from not from your parents, this is actually you. You're actually funny because your genes are just not from, this is not followed from, this is not a nature, this is nurture, something like that. And also, what you were saying, the same thing that has been published here, look at this, nature and nurture. Exactly what you guys are doing here. This is exactly what it is. This is from the genetics from your parents, this. And this one, this is this, not from your parents. This is you, because you're going and talking to this in the park and sitting there, because this is not your parents. This is you, don't want to study, you have other friends, and you just have to, you have other girlfriend or someone is sitting in the, bench and talking nonsense and all things like that. So he was giving us a real picture of how to study. Means that take the one that is going around you, what you are, and think about it. It's actually that is what is the science. So science is about, the biology attracts everyone. You're all biologists kind of, because it's actually happening, right? It's not physical phenomena. It's happening, and that's why you're studying. You're attracted to it. That's what I think motivated us, that he taught us by taking the research articles, not by textbook, not by anything. That research is actually changed a lot of kids, not just me, many of them in our college days. So it's, I think, as a teacher, is very important. How much you learned, and how much you are learning, how you are actually teaching to a next generation. If you are not passionate to teach, you should not take up the teaching. Okay, you had to have the passion to teach. So that those teachers are passionate completely. I mean, that is overwhelming. Actually, not just me, everyone who actually studied there, zoology. And like that, there are many other teachers in many other classrooms, right? There was a Padmaji, he is a chemistry teacher. He was actually always with a suit every day. It doesn't matter summer or the winter, because that from there we learn the discipline. Like, because he learned, he went outside the world, country, and then learned and came back. He was always disciplined, perfect, and you don't want anyone to be that, he's pin of silence, everything. So is this discipline you learn from him, there's so much, like his name is Dr. Padmaji. So like that you all learn from each teacher something. So I learned those things probably. And when I went to, when I wanted to become a doctor, I think I wanted to do the medicine. But believe me, during that time, that year, the entrance exam came in Mysore University. That was the first entrance exam. There was a Kaveri Galata, and then everything was happening, and then entrance exam was postponed, and then postponed. We had like long holiday, and then finally entrance exam happened. That's the first time entrance exam, because there was a fighting. There was like not to have the entrance exam for masters. So the entrance exam, then I cleared. I was cleared MBA. I was in the botany and the zoology, and then when I got into a botany, this is a story. You asked me story. I think important, because you all have a story to tell for sure. And this is my story, and probably you will have your own story. Probably you will be even fantastic. Yours will be probably when you become someone. I think probably start talking about all your background and everything. So when I got into botany, and I wanted to go to zoology, because I had a professor, Dr. Rangnath, who is a NAC director, and I was related to him. So I was former NAC director. So I wanted to really do zoology, because the zoology attracted to me during my undergrad. And somehow I did not get it in the first list. And so I got in the botany, and then I joined botany. And then I was like, not disappointed a little bit, because zoology was attracted, right? And then during that time, I was in NCC, and then I had a NCC during that time. Even my BSc or NCC, I was a C certificate, I was an under officer, and also I went to a lot of integrative camps, and then military camps, and BLC, and a lot of things. I did a lot. So that's why I did not study so well. So I was like doing a lot of other integrative. I went to a lot of places, military attachment camps, and leadership camps, and many things. So during that time, I did get a lot of scores. Then went into NCC. I scored a good number, and then through NCC quota, I got into biochemistry. So because biochemistry in Mysore University during that time, 20 seats, right? There were no other biochemistry in the all over Mysore University. There was only one department. And in biochemistry, I think 20, most of 15 people will be rank students, all of them rank students. And then we have 5 from the other categories, and sports quota and other things, foreign quota and things like that. But it was tough for general biochemistry students to do the biochemistry. It was not easy. My own friends were there right next to me. It's like tough, because they are from the BBM, biochemistry, microbiology, but I'm from the chemistry. So it was challenging. So we did study though, but it was very challenging for the general chemistry students to do the chemistry, biochemistry. But we had great teachers. Okay, we had great teachers, and T.V. Veerabhasapagowda, and then my MKK is, and then many teachers. There were a lot of Cletus D'Souza. I think he's from Mangalore University. He was also my teacher, and many more. Okay, the great teachers, I think, changed our way of thinking science, right? Way of thinking the research. The Veerabhasapagowda, TVG we call. He was a professor, and he guided me during my master's thesis. And I think there are a lot of things we learned. A lot of things we learned. And from chemistry, I think teachers were attractive, because Rangappa and Timaya during that time. There are many other chemistry teachers after that, and zoology. Many Ranganath is one of the geneticists. I think. I listened to him. He gave me one book, genetic book, to read and study during that time. I still remember. Like that, many teachers were there, just not them. The great campus, attractive campus. But important thing is that I think key factor when I was in bachelor degree. I know Joshi knows. I think I used to go to Mansa Gangotri, that is a campus, PG campus. I used to study in that library. So it means that somehow I had a librarian attached. Knew these. Through someone I used to hide and go and study, because I wanted some peace area, environment. We were in the small rooms, and very difficult. There were 3-4 people in one room, and things like that. And during that time, cooking and all those things. So important, I think, this. If you want to really study, you will find a way. That's all I can tell you. So I was also Kannada literary. I used to read a lot of personalities in Kannada language. So I used to go to Kannada area, where if you go to Mansagangotri, there is a left side if you go, there's a Kannada and section and then history and all those. In the right, if you go to science, and there's a, I think Joshi might have studied there also during his exam time and all those things. So that's how the life is. After I think masters, you don't know what you want to do. Believe me, you want to become, probably we all wanted to become a medical rep or things like that, because it was tough even during that time to get a job. Then we all end up in CFTRI, become research assistants. That's what we did. Everyone, become a research assistant because there are a lot of projects there, and that's what I did too. And but I felt that I'm wasting a lot of time, because by the time you have a master and your parents are expecting something from you, right? And you had to get settled or get job, right? But I was not doing that. That is like 1,500 rupees is not enough. 1,200 rupees or something. It was not enough after masters, right? 1,000 rupees is like, just give a room rent, that's all. And so that actually, I went back and told my dad that I wanted to study more. I just cannot do this, because without studying further, I don't think I will be able to do anything, okay? So my dad said, even at that time, even the difficult time, he said, who asked you to go and work? I did not tell you to go and work, but go study whatever you want to do. Then I started studying for competitive exams, did a lot of, there was a wing commander Raghunath. He was a great teacher for me. He was a army guy and who actually fought in Germany, and he was a, Mysoreans know him very well. And even to go to his classroom, he's a private classroom, free classroom for a competitive exam. He used to teach history. I never learned history when I was in school time, but I learned there after my masters, history, geography, understanding the whole concept of historians and their lifestyle, and how important it is, and also personalities, I think, development. Someone like him, I think, a big difference in your life, right? There was a Narayan Swami, G. Narayan Swami is well-known commissioner. Now I think he's placed in somewhere else, as he's a commissioner at that time in the income tax. And he was a botany MSc, and he did IRS. He was actually guiding us for essay writing, also competitive exam, anything like AS, coaching. Something like coaching means that talking about, it's not that you can, they see, one thing I keep telling, even science, I cannot change you. You have to change yourself. Same thing. Narayan Swami used to tell me that you have to change yourself. Only he can actually guide you. Okay? He's such a great personality. When we go to his office, even though we were like masters kids, he used to give us, first thing he used to do is order tea for everyone, all the students, and after the tea, he used to discuss what you have written essay writing. For example, he will give you one of the writing, what you have to write, and then we write, and then we go there. That's how our writings are improved. Even though I do scientific writing, something that it actually always, you think about it, what the changes that you learn during that time, right? That it is for life. In life, you just keep coming back, right? So this is a life lesson. Once I started studying, I think I started clearing UGC, any exams I take, any entrance exam, any competitive exam, because you get that confidence, right? Some kids, I think it's very difficult to get that confidence. Some kids, they do it just like that, right? So for some kids, like me, probably I needed that whole environment after my masters. So that changed the way I think about competitive exams or any exams are going to the interview. So after that interview, I cleared and went into DFRL because I wanted to go to be local, because I was studying for the competitive exams. So and it helped us a lot. But as you enter the scientific world, when the competitive exam did not go through the way we think, right? Because as an background, what our background is, and also having a family with 4 kids and things like that, and having a fellowship, DRDO fellowship, even 2,500 rupees is a great thing at that time. I had a lot of money. I didn't even buy a bike, in a loan. What I'm saying is that 2,500 is a lot. And that was actually changing, right? From 1,000 rupees to 2,500 is a lot of money. And then the PhD, I think having a right mentor. My mentor was a ISC PhD. I think he was a DFRL director at that time. He was a department chair, biochemistry, and then that actually mentorship, I think a little bit. Dr. Santanam, he was a great mentor because of the experience. He went to US and then had a postdoc and came back. So yeah, he knew the world, he knew what science is, and then he's from well-trained institutions. So the mentorship, I think you have to have a mentor who actually have been trained well. You have to find a mentor who actually went outside and world and has seen a little bit here and there. Having a right mentor changes you a lot, changes your life too. So at least whether in mentoring or not, the personalities is a key thing. So what type of personality the mentor has is a key factor for your life to change. Because whether they're going to teach you or not, just having a personality is also important. So that type of personality, even today, I just messaged yesterday to my mentor. He's in Bangalore, so I'm going to meet him next few days. But important, whenever I come, even when I was giving any lecture anywhere, he comes there and he is so happy to be there. You know what I'm saying? So it's about that type of mentor is a key factor. Like I'm coming from somewhere and giving a lecture. When he comes to Boston, his kids are there. When I go to Boston, I met him one time, and he was so happy. So it's important, that relationship that important for your life. Whatever the mentor is, that personality is important. Keep that in mind. That it doesn't matter your stage now or later. I have a mentor through all my career. Even now, we have a mentor too. Mentor means that someone is guiding you through something. There's always, you are to find a mentor every stage of your life. Is a key factor. Right? It's not that you're not over. Even though you're a professor, you have to have a mentor who actually guide you to the next level. You can actually guide you what else you can do. It's not over yet. You know what I'm saying? So those kind of relationship and important. After that, I think I gave you already hint at how I did my research and what difficulties were there. And even though there are many difficulties, I was at DFRL, we had a lot of problems too during that, because it's a defense laboratory. They didn't know the research, JRF, what is the JRF means that in the defense environment, you guys may not think, because I know what it is in a defense environment, because during that time it was like that, but now it has been changed. And believe me, there are a lot of researchers coming out from those institutions, and DRDO fellowship is a great fellowship. Like any other fellowship, any CSIR fellowship, it's a competitive. So it's that institution is changing, okay? And you need to have the institution, university is also important, and for a base. And you don't have to learn anything from it. You have to. End of the day, it's you. You change and you want it, you will get it. If you don't want, you will never get it. Okay. And if you blaming anyone, again I'm repeating, so you'll never get it. Okay? You just have to change yourself and keep doing what is right for you and your life. Okay? So end of the day, you change your life, no one else will change yours. So that's what I think I learned, those tips on the way. So I really wanted to do research. That's the reason that I went to US, and I think there are a lot of tough times, believe me. It is like it looks great from outside. It is not like that. Okay, there is a lot of tough life out there too. Okay, there is a lot of crying. There are a lot of struggle. There are a lot of you go through, and there is the food issues, and you have to cook. Otherwise, you will not get the food, and you have to cook yourself, and you cannot eat burger every day. And you want to go to burger shop, and there is a culture shock. And we learned all those things, right? Many days there was even we didn't know whether there is a hot water and cold water in the shower at that time. You know what I'm saying? I was always taking cold water. I didn't know there was a hot water also other side. things like that. But my aim was to do cancer research. I would do research, something to do. But my intentions were not anything else. But I knew that I can go anywhere and I can survive and do. You know what I'm saying? I took like I think $100. That's all when I went to US. That's all I had in my money in my pocket. My mentor gave me a check. My mentor gave me a check. She was like, really, you have like this much money and you want to survive in US? I was like, she was shocked. And then and she gave me a check. And then I had to write a signature saying that after my first salary, I'll return the money. So that's how I think Eugenia Wang, her name. So she gave me that first check. And that's how we have survived. So, okay, if you don't have money also, you can actually survive. You don't need, there are people, but you have to believe in yourself that you can do it. If I were to say, like, I don't have money, I cannot go to US, somehow, you know what I'm saying? So, I went and so we survived. I didn't know anything about anything in the world, okay? I didn't know how to, first of all, I didn't know there's a hot water, right? Think about it. I didn't know anything, okay? So, but we learned. But my focus was this, research. That's why I went. There's nothing else, right? So, on the go, you learn everything. Either, cooking to anything, living and having friends. And having friends like, someone from an institution like IASC is a great thing, right? You get everything there because it's hard to go get it here. When you go outside, they all become friends and you learn from them and you share yourself and they share. Initially there was a struggle. There were other people who were publishing great papers. I was not. You know what I'm saying? But what do I do to get those type of paper? I left the university, went to Vanderbilt. It's like a Harvard University when I got a chance. Because to do better science, to do more models, do more in vivo, translational, things like that. You have to challenge yourself. There is a lot of other jobs come in between. I would have become an assistant professor. I would have become something else in there also. I could have gone to food nutrition biochemistry. I would have become assistant professor. That was not my intention. The intention was to do cancer biology. So I had to survive, strive, and go to other universities, learn, and struggle. And again, you know what I'm saying? There are a lot of family struggle, life struggle, and then a lot of things. There's story. A lot, 25 years of story. You want to hear? A lot. Okay, there is struggle everywhere, every day. And end of the day, I think you all will be happy. So we are all happy. Doesn't matter how much the struggle we go through, right? So you all will be happy. I think you all will see the happy ending, right? It's important. You all see it. I think you should not forget one thing is that who train you, who guide you, and like I was saying, like my mentor. Like, I always see him and try to talk to him. It's important because even I become mentor too. I do the same thing to my next generation. It's the same thing. This is important when someone is teaching you, and you should not forget. And also someone, your students has to be like that. So you have to train them such a way that humanity exists and will be there forever, right? So that's the story. Thank you. ಸ್ಟಾರ್ಟ್ ಮಾಡೋದಾ?
It's not about perfect, it's about the effort. And when you bring that effort every single day, that's where the transformation happens. A very good afternoon to everyone. On behalf of Mangalore University Biochemistry Program, I feel both honored and privileged to have you all here for the valedictory function of Global Initiative of Academic Networks GIAN program on developing cancer model in vitro and in vivo advantages and limitations that has been going on from 18th March 2025 to 22nd March 2025. Today we have Professor Manjaya, Dean Faculty of Science, Dr. Nagaraj Nagatihalli, Assistant Director for Basic Science Research and Associate Professor of Surgery, Pancreatic Cancer Center, Sylvester Pancreatic Cancer Research Institute, University of Miami, Florida, USA as a resource person, Professor Ganesh Sanjeev, Local Coordinator, GIAN Program, Professor Chandrasekhar G. Joshi, Coordinator, Biochemistry Program, Mangalore University and Coordinator of GIAN Program. As we commence this event further, it's our duty to respect and welcome our guests and all present here. So I request Dr. Lekha, Faculty Department of Biochemistry, Mangalore University, to formally welcome the gathering.
Good afternoon to one and all gathered here. It is indeed an honor and privilege to take a moment and extend a warm welcome to the valedictory ceremony of GIAN program. As we gather here today, we reflect on the incredible journey we have undertaken over the past few days, a journey filled with insightful discussions, enlightening presentations and valuable networking. It's my pleasure to welcome our distinguished resource person, Dr. Nagaraj Nagatihalli, Assistant Director for Basic Science Research and Associate Professor of Surgery, Pancreatic Cancer Center, Sylvester, Pancreatic Cancer Research Institute, University of Miami, USA. I request Professor Sarojini BK, Senior Professor, Chairperson, Department of Industrial Chemistry, to florally welcome Sir. Thank you, ma'am. I would like to welcome Professor Manjaya D.H., Dean, Faculty of Science, for gracing this occasion. We are fortunate enough to have you, sir. I request Professor Sripada K.S., Chairperson, Department of Zoology, to florally welcome, sir. Thank you, sir. I would like to extend a warm welcome to Professor Ganesh Sanjeev, Local Coordinator of the GIAN program, for his commitment and support in planning the program. I request Professor Monica Sadanand, IQAC Director, Mangalore University, to hand over the bouquet to Sir. Thank you, ma'am. I would also like to welcome Professor Chandrasekhar G. Joshi, Coordinator of Biochemistry Program and GIAN Course Coordinator, for his persistent efforts in organizing this event. I request Professor Chandra M., Department of Biosciences, Mangalore University, to florally welcome Sir. Thank you, ma'am. A heartfelt welcome to all the participants. Last but not the least, I would like to welcome all my colleagues, non-teaching staffs, research scholars, students for their hard work and commitment in organizing this program. Finally, I would like to welcome each one of you for gracing this occasion with your valuable presence. Thank you.
Thank you, ma'am. I welcome you too. Esteemed participants, as we draw this workshop to close, we would like to take a moment to request your valuable feedback. Your perspectives are crucial to us in ensuring we continue to deliver meaningful and enriching experiences. So now, I request the participants to give your opinion.
Good afternoon, respected delegates on the dais and also off the dais, and also my fellow participants. I really so much of happy and also lot of knowledge I gained through this GIAN workshop for the period of 5 days. So maybe though I have undergone 1 year CV Raman postdoctoral fellowship way back to 2013-14, and also I did so much of my work, postdoctoral work, is in cancer biology, that too in pancreatic cancer also. So despite of that, so it is my interest to attend. When Joshi sent a brochure of this workshop, immediately I approached Joshi, so kindly register my name for this workshop, because I need to learn, I need to update what are all the advances which are happening in the field of cancer biology. So cancer biology is itself is a ocean. If you go to US, so many laboratories, and about 80% of the research work in US, it is going in the field of cancer. It is such a ocean. So therefore, so maybe I did my postdoctoral studies way back to 10 years back, almost 10-12 years back. Still there is so much more advances might have been happened. So really that is after going through the lectures of these 5 days, I came to know that lot of advances have been happening. So maybe, so this workshop, we have a knowledgeable person, a resource person, and also a competent personality we have today. So that is Dr. Nagatalli Nagaraj. The way in which he has explained, so starting from first day to last day, he has got that the same tempo, we need to appreciate that one. And the energy he has, and there is no pause between whenever he is explaining the things. And he taught us, so starting from basics of cancer biology, by taking a suitable examples of in his field, in his own field, almost he strived so many years, and so many, maybe 25 to 30 years in the field of cancer biology. That too especially, so pancreatic cancer. So maybe, so we are very fortunate to have him here today. So always when I attended this kind of lectures, workshops, so I came to an anecdote in my mind. So you might have all heard about Subramaniam Chandrasekhar. He is a Indian born American scientist. He is a great astrophysicist. So he got the Nobel Prize way back to 1983. So he is such a wonderful scientist. So maybe he was working in some Chicago University. So in those days, so each professor used to offer some open elective, like in our system, CBSE system, we have an open elective. So many students sometimes, so university will offer, different departments will offer many open elective courses. There will be sometimes among all those open elective courses, there will be one course, it will be catching all the students. So there will be huge rush for one student, for all the students. Likewise, so Subramaniam Chandrasekhar, he was also offering an astrophysics as one of the course, open elective. So in those days, many other professors, those who are also offering open elective courses, but for other professors open elective courses, many students have been enrolled, signed up for open elective courses. First for Subramaniam Chandrasekhar, so who offered this astrophysics, hardly 2 students they signed up for this course. Then all those professors in those Chicago University, they were ridiculed and they mocked and they are kindling and they are looking in a different way about Chandrasekhar, Subramaniam Chandrasekhar. So but Subramaniam Chandrasekhar, he never cared. He never bothered about what others they will teach. And all those other professors, they will think that, so now Subramaniam Chandrasekhar has taken, for his course, only 2 students have been enrolled. They think that he will going to cancel this program. But so that Chandrasekhar has got such a enduring dedication towards education. He is very fond of his students and is very loving about his area, that is astrophysics. But for all the students, see, this course was happening in different area. That is about 250 kilometers from the main campus. So every day, weekend, he has to travel 250 kilometers and again 250 kilometers to teach only 2 students who have been enrolled for astrophysics. But luckily, see, look at that. So he is all the way driving 250 kilometers every weekend to teach only 2 students. But think that, so he is so passionate about his teaching. He wanted to develop the knowledge and inculcate the knowledge of astrophysics in those students, only 2 students. You may surprise, those students, those 2 students, they got Nobel Prize in the physics. Before he could able to get the Nobel Prize. See, the students, they got Nobel Prize early compared to his own teacher, Subramaniam Chandrasekhar, who got the Nobel Prize way back to 1983. So that is what, see, Subramaniam Chandrasekhar has traveled only 250 kilometers. Now today we have Dr. Nagathelli Nagaraj. Such a wonderful personalities and inspiring personality and also knowledgeful personality. You may not believe how many kilometers he has traveled. He has traveled approximately about 12,000 to 13,000 kilometers from Florida to here. Maybe it is about 20 to 25 hours of journey with lot of dedication, and he has got such a zeal, something I need to give the society. And really, I really admired all his personalities and also the lectures he has taught us. Maybe all the youngsters, young minds who have gathered here. So maybe some of them are MSc students and some of them are already enrolled PhD. So you all cleared your all doubts how to design your research. Sometimes you feel that, sir, today you have to write a project. Today you have to write a project. So he taught us, so what is meant by a literature survey? What, how best we have to do background and literature search. And also we have to think that what is the uniqueness in your PhD title and what are all your objectives. Once all these things will be done, your 50% of your PhD work will be over. So maybe, so that is what Nagaraj sir was telling so many times. Keep on is pressing us that yes, you have to prepare in this way, you have to develop and you have to instill on your own. So that is what self-motivation is the best motivation. So that is what I really fortunate to attend this kind of workshops. Definitely we need this kind of workshops. So maybe this will again regenerate our what is called as interest and also so focus also. So lastly, I don't want to take much time. There are many students, they want to share their experience. So always there is a proverb in China, it's a Chinese proverb. That is, if you want to plan for 50 years, we need to plant a rice. If you want to plan for, so 500 years, you have to plant trees. Suppose if you are planning for 1000 years, we need to educate the people. So that is what, so with the intention of preparing all these younger minds for 1000 years, so we are very lucky to have, so Nagaraj sir here. So he was there with us for these 5 days. Really, so we interacted well. So all the lectures, suggestions, and what are all the techniques, maybe it will be difficult to understand all the technologies he has used. Knockout mice, it is new to us, especially in Indian scenario. And then siRNA, shRNA, CRISPR-Cas9, these are all advanced technologies all our students should know so that we can understand. Otherwise, little bit difficult to understand, especially mass spec analysis and PET imaging, all these things. See, all these things I know, so when I had been to my post-doctoral studies. Otherwise, I wouldn't have been, I would have missed so many opportunities. See, that is how you have to go out. So this, these kind of lectures, definitely, which will bring lot of confidence in all the young minds. So I also thank Dr. Joshi for organizing this kind of workshop. See, they did a lot of, so they took so much of time. So this workshop, though it has been happened in only 5 days, but for execution of this 5 days workshop, they have struggled 1 year. So they are in constant touch with almost 1 year to bring out this outline of this project, that is this workshop, and they submitted to GIAN and they presented. Ultimately, it got accepted. Really, I congratulate Joshi and his team and also the Mangalore University administrative authorities for supporting him to conduct such a nice, wonderful workshop. So thank you, sir. Thank you. Thank you.
Good afternoon, everyone. Respected dignitaries on the dais, off the dais, fellow participants, student friends. First and foremost, I would like to thank GIAN program, Mangalore University and Professor Chandrasekhar Joshi for organizing this such a wonderful program. As many of participants, some are still PG students, some are scholars, and some are faculties like us. It is not easy to access a pioneer in the field of cancer biology who want, as we want to learn about cancer biology. We study cancer biology in our academics, but it is very different what we learn in academics and what we are doing and what they are doing in the research. As sir told, they are in the verge of using recent advanced technologies that we don't know, and it is very difficult to access those technologies. And as sir told, wherever you are, it is just a focus. You need to focus on your work, even if you have the access to the technologies. It's you what you want to do and where you want to do and how you want to do. In all these 5 days, the main focus and what I learned from sir is, don't care about where you are, what you have. You just need to find a way you want to do. So that is the main take home message I think I have to, and everyone has to take. Wherever you are, you just need to focus. You will get one or the other way. You need to collaborate or anything else. You just need to find a way to do that. You just need to focus on what you have to do. You have to think about a long-term goal where you have to reach and what you have to do. Based on that, you plan and you focus, talk to them, talk to other researchers. That is the main take-home message apart from cancer biology what I learned in these 5 days. And as I told you, I would be very grateful to Chandrasekhar Joshi sir for organizing this, to interact with such a great scientist and pioneer in the area of cancer biology. I came here as an, it is a stepping stone for me to enter into the world of cancer biology. We have learned about cancer biology, but as I told you, it is different, that is academics and research. So as I have the intention of starting a research in cancer biology, so I came here as I talked with the Joshi sir also. So is it good for me to attend here and can I learn something? And he told that it is a great opportunity, you come here. So I grabbed this opportunity, I think, and I have learned a lot. And as sir told, it is a great thing to start from here. Whenever you are, wherever you are, you start from now itself. So I think it's a great experience for all of you. I think participants know, and it is a lifetime experience for me. And to initiate, and it's a great, as you told, no one will help you. It is like they will guide us. Today itself, he told that anybody, no one will help you. They just guide you. So it is a great guiding experience for us, for me, to start my work from here. Once again, I would like to thank the organizers, Joshi sir, GIAN program, and at last, I would like to thank the great scientist who came all the way from Florida to talk his experience, tell his stories, and it is not possible to interact with him for 5 days. It is difficult to talk to him for 5 minutes for us, isn't it? So it is a great experience to talk, just to live with him for 5 days. It's a great experience. Thank you, sir. Thank you so much.
Thank you, participants. Today we are very honored to have Professor Ganesh Sanjeev, local coordinator, GIAN program with us. Now let's all warmly welcome him to share his valuable insights. Over to you, sir.
The Dean of Faculty of Science and Technology of Mangalore University, Professor Manjaya D.H., the star of not only this concluding ceremony, but the star since last 5-6 days and some more years in your mind, Professor Nagaraj Nagathelli, and the coordinator of this GIAN program, my colleague, Professor Chandrasekhar Joshi, my esteemed colleagues from sister departments and institutions, the delegates, the participants of this GIAN program, ladies and gentlemen. Very good afternoon to everyone. GIAN program was started in the year 2014-15 by the central government through the then MHRD, the Ministry of Human Research Development. And in the initial phase, we could get a good number of programs sanctioned by the government. They made IIT Kharagpur as the nodal institute for all the national coordinating institute for this GIAN program. First, second and third phase, IIT Kharagpur was the nodal institute. But after third phase, there was a changeover of institution from IIT Kharagpur to IIT. Fourth phase, there were some fusions among both the coordinator side as well as some institution side. You may be knowing that there are around 450 state universities in our country, apart from private or deemed to be universities. Out of these, only 24 institutions were selected in the beginning. We were one of them during those days. And till date, we have been sanctioned with 30 GIAN programs. We completed first, second, third phase, we completed 24 GIAN programs. Actually, 26 were sanctioned. 2 we could not conduct here because one of the staff members got, and another one because of corona. One expert from UK, because of his health problem, he could not come over here, not even conduct that program online also. So 26 sanctioned, 24 completed. In the fourth phase, we had submitted 17 proposals, but nobody knows where these 17 proposals were routed to, but only 2 proposals were through in the beginning. Now again we submitted some more proposals. So under fourth phase, we could get around 4 programs sanctioned. So totally, this is the 28th program running today, and 2 more are in queue, maybe one in computer science and another one in physical education. So this program is a beautiful program to tap experts in various fields of research from international community, because there are experts all over the world, and you can tap the most innovative or most updated researcher, so that you can get their interaction here through their lectures, and also you start collaborating with them. You can get scientific support or whatever knowledge level based help from them, so that we can always enhance the level of our knowledge. And one more important thing I want to tell you here is the students, especially the participants who attended this program, because so many programs have been conducted by so many institutions. In our state, especially in Karnataka, only 3 institutions were selected for this. Karnataka University Dharwad, University of Mysore, and Mangalore University. We could get this much of research programs, because I don't know why they have not tried for many programs. They had only around 4 or 5 programs in other institutions. They could have tried actually, but we have conducted so many programs, and the students or the delegates, the participants who attends 1 week program, they get 1 credit. Nowadays, under NEP there is academic credit of, academic bank credit. So you can always dump this credit there. So if you are attending so many programs, you can dump all these credits there. In case you want to do some programs in IITs or some national institutes, if the syllabus is matching whatever you have studied already, under this GIAN program, you can shift that credit directly to that particular institution, and you need not attend those classes in that particular institution. Automatically, you need not attend that exams also. So you keep with credit, whatever credit you got, you update in your ABC account. I think almost the students are all having ABC account, so you can always update it. And I think staff members, researchers, if you are interested, you can also open it with the permission of our NEP, your respective NEP nodal officers of your respective institutions, and you can discuss with them how to open an ABC account and keep this credit in that particular ABC account. So this is the advantage. Plus, for students, you can always talk to sir whenever you want. I think we have had so many lectures by GIAN experts. All of them are having contact with our colleagues now. So even the students who attended these programs, they are sharing their, they are expressing their whatever knowledge they have acquired from these experts, and they are seeking some help from those experts, and some of them are doing good piece of work in their respective laboratories. So make use of this opportunity to excel in your life. And Professor Joshi has arranged, I think, each and everything to you in a befitting way, and if any shortcomings are there, we take the responsibility from university side. I am not telling that he is not from the university, but he has put a lot of hard work to organize this, and I congratulate all of you for attending this GIAN program, and you will be rewarded with your respective grade points. And more than that, I thank Professor Nagaraj Nagatili for accepting our invitation and for being with us and sharing his knowledge, and also extending hand to help you all. I thank one and all for being here and making this GIAN program a successful one. Thank you one and all.
Thank you, sir. As a token of respect and appreciation, we would like to felicitate our resource person of GIAN program, Dr. Nagaraj Nagatihalli. I request all the dignitaries to do the honor. Before that, I request Dr. Nivedita NB, Faculty, Department of Biochemistry, Mangalore University, to give a brief introduction about our expert.
It's our privilege to honor our distinguished guest, Dr. Nagaraj Nagatihalli, a leading expert in cancer biology and translational medicine. Dr. Nagatihalli currently serves as an Associate Professor of Surgery and Graduate Faculty in Cancer Biology at the University of Miami, Miller School of Medicine. He is also an integral member of Sylvester Comprehensive Cancer Research Center, where he leads groundbreaking research on pancreatic, lung, and oral cancer. Dr. Nagatihalli holds a PhD in biochemistry from the Defense Food Research Laboratory, Mysore in 2002, following which he pursued postdoctoral research at the University of Louisville and Vanderbilt. His academic journey has been marked by a commitment to unraveling the molecular mechanism underlying tumorigenesis, drug resistance, and targeted therapies. Over the years, Dr. Nagatihalli's work has earned him numerous national and international recognition. Some of his most notable honors include Young Investigator Award from the American Pancreatic Association, Stanley Glasser Research Award, University of Miami, James and Esther King Biomedical Research Award, Florida Department of Health, multiple travel scholarship from organizers such as National Pancreatic Foundation, the American Association for Cancer Research, and the Collaborative Alliance for Pancreatic Education and Research. Dr. Nagatihalli has been highly successful in securing competitive research gratitudes with 8 active grants and 16 pending grants, including NIH, RON grants worth of $1.7 million, which is around 13 crores in Indian rupees, Florida Health Research Grant worth of $0.58 million, which is around 5 crores in Indian rupees, NIH RON worth of $2.48 million, which is around 21 crore of Indian rupees. His research has also contributed to phase 1 and 2 clinical trials targeting key pathways in pancreatic cancer. Sir has published extensively in high-impact journals with over 60 peer-reviewed research articles, 4 book chapters and 127 papers presented in various conferences. He is also contributed for various editorial boards such as Managing Editor of Nutritional Journal, Associate Editor of Frontiers in Gastroenterology, Associate Editor of Frontiers in Oncology, or Editorial Board Member of for Journal of Biological Science and many more. He is also reviewed for various national and international research grant proposals. Beyond research, Dr. Nagathireddy is a dedicated mentor and educator, having guided over 15 postdoctoral fellows, numerous graduate students and medical residents. We immensely thank you for being with us and sharing your knowledge with our young enthusiastic participant. Thank you, sir.
Now, I request our resource person, Dr. Nagaraj Nagathihalli, to enlighten us with his valuable thoughts. Over to you, sir.
Thank you so much. Honorable, and I think Vice Chancellor, he was not here, Dr. Dharma and Dr. Joshi, sir, everyone, delegates and also professors, faculty and members, students, also participants of this GIAN program. Thank you so much for this lovely last 5 days. And I think this is so important for Indian students to have this type of experience. That last 5 days, what we have been through and what we have discussed, interaction. I think this is amazing because we, myself and Joshi, when he was postdoc coming to US, I don't say that in my laboratory. We wanted him as a colleague. I wanted to share all the research knowledge what we had when he was an assistant professor. I think that's when I think he came. Important is that even before him, there were 2 other faculties came to my lab as fellows, like an Obama Fellowship, an international fellowship, to the laboratory and learn a lot of techniques, and also a lot of other investigators that came, like in a weekly program and 10 days program and in the laboratory. The reason I wanted to have them there, it is not easy, because to have them there and also spend the money and also my time and the others' time and the resources and what they go through every day. It's not easy, but important to do. This is what we learned all these years, to share our knowledge, and also one or the other way, and also help others one or the other way, whatever we can. I think that is what I think we were doing. It's also, this is a learning process. It's from my parents, and I think that's what my dad used to do, and I think probably carried over. That's all. It's a genetic thing. Okay. So when Joshi was there, when we came back, and we were in touch for a long time, I think many years. We always talked about. He was always asking me to come and visit Mangalore University sometime and all. So I was always telling him that visiting and giving a talk is not going to be enough for our students. I think something we had to do some program where they can actually interact and get knowledge, and do something like much better than just giving a talk and going. So this is what I think. Same time the GIAN program came, I think he was actually excited. He contacted me, and we wrote together. I think he actually had a framework, and I think we did it and we got it. So when we got it, when he got it, he messaged me and said, "Oh my God, we got it." So what do we do? So well, we have to do it. And how do we do it? It's things like that. So it's a great program. The reason is this. Okay, cancer biology is vast. Like there is doesn't matter. We always think that we learned something. There is always new thing comes, and every day, other day, there is a publication. The high impact paper, what we have been through last 5 days, and you have seen how much technology has changed, how much knowledge has been changed also in the field. Every other day there is a high impact paper with so much of information, and our students are getting diluted with that. Not able to focus, not able to do. They think that they are low. They just cannot be done. So this is what I think I wanted to give the energy to you, that we have done the same way. We were in the same situation, but we have to take one by one, cut everything. We are in the top-notch research in the laboratory in US. The reason I'll tell you that Joshi was there. One of the top-notch pancreatic cancer research we are doing in my group. The reason I'll tell you why, because we are on it all the time. And there is any paper comes out, we will be on it. We were going to discuss that paper, what they are doing, and even we go to conferences, we'll be on the top-notch researchers. We'll be searching for those researchers' publications or the presentations, and we will be on it. We will discuss with the postdoc and grad students, and then we will not leave any technology. If we need money, I'm telling you that we will sit and write those grants all the time. And we never had a money issue. The reason I'll tell you, because we will write the grants and we will challenge the government. We will challenge whoever is NIH or Department of Health or many other organizations, American Cancer Society, AACR, any organization, wherever the money is, we will be getting it. So that is why our technology-wise, I think students are probably overwhelmed with all the technology we were using in the laboratory. Anything that you name it, we have it, we were working on it. Or we have a plan. So that is how it is. So why? Because we have to do it. If the Stanford, I was telling you today morning, if the Stanford, Harvard, and MIT or Pennsylvania University or Vanderbilt University students are coming to my laboratory and trying to do the research, or even the MDs, clinicians are coming to my laboratory doing the research, means that there is something. That's why they are coming. So it doesn't matter. I'm a village boy going all the way, to US. That's what I was telling. That's my story to you all. The reason is that if I can do it, you can do it. And anybody can do it, anything. And if you think that you cannot do it, start today. I'll tell you, after 5 years, you will see yourself where you can go. But every day you have to work hard. Sleepless, work hard, and you can focus and challenge the world. I think you can do it. You will do it. And we all did it. That's what I keep telling Joshi. Sometimes it's overwhelming because he always tell me a problem. That just not Joshi. There are my colleagues, my friends, they are here working in the research laboratory. Even in IAC, my friend professor also give me a lot of reasons why we will not be able to do it, why we cannot do it. I was telling you, Vemala from INSTEM, that I have a friend who is actually tell me, he's the best chemist we have. and he's telling me a lot of problems. But even though we have these problems, you know what? He is one of the pioneer in the chemistry, even from India. He from Ganesh is published fantastic genetics paper and DNA damage repair mechanism studies. Is a professor there. What I'm saying is that even though there is always lacking here and there, believe me, if they want to do it, they can do it, because they have been trained in Harvard University, both of them, and they know what it takes to really publish a great paper. It is hard, but they work harder than hard. That's why they were able to publish from here. And if you think that we will not be able to publish in ASCR Cancer Research Journal, no, they all published it. You know what? Why they published it? Because of not the tag of the Harvard University they had. They are that learning, that whatever they have learned, and that tough learning process, right? They know they have to do it, means that they will do it. Day and night they work hard, they will do it. So thanks to everyone for being there and silently getting all the inspiration or knowledge and the science that you wanted. I think this is needed for you all. That's all I can tell you. Because I tell my students too, my postdocs, they come to laboratory weekend and they don't know what to do. I know them. We were all there in the same situation when we went to these countries. And we went to those countries. The reason is that we want to do research. That's why we went to country. We don't want problem. We want solution. I want to bring the solution to the field, I don't want to talk about problem, that's why we went. It doesn't matter where we are and what we publish, I keep telling, it is for the humanity. It doesn't matter, my publications will be there everywhere. My clinical trials and anything knowledge that have been created has been used in many other, anybody can use it anywhere in the whole world. But we wanted to challenge the field and the researchers and want to go and fight and get it done. That is the reason that we were there. So I actually can tell that you definitely, most of you will do great in the future. I think this is something that it doesn't matter whether it's a cancer biology, plant, food, and I keep telling food technology or any other genetics or pharmacology, or doesn't matter which area you go to, you will do great. Okay, but important I think for our students is that they all need something like like this hit. I think this is what we also needed and we also got. I got it from Wing Commander Raghunath. I was telling you about our ARS officer Narayan Swami and many Professor Panduranga, Professor Ranganath, and many of them. On the road we got all these tips. My NCC officer, okay, is a Swami, JSS Swamiji's brother, is Dr. Manjunath. It was like inspiration to us. So it's all something that we have gained, and I'm just telling you all these tips. The reason is that I think today is a day for you to go back and start thinking about your future for better, right? And you all get, when my postdoc says, do you, sir, if I do this, then my salary will change. Yes, you publish high impact paper, your salary will definitely change. You don't need to run behind the salary. I have a better salary today. The reason is that I'm running behind the science, not behind the money. So you have to run behind your work, not behind the money. Probably in this Indian scenario may be different, but believe me, it doesn't matter whether India or US or any other country. I think if you do your work best, someone will be always watching, and definitely there will be something changes in your life too. And you can be whatever you want. Today money is, I have enough money, but money is not the key. Key is that my focus has never changed. Like, Sir was saying that I have a same energy because it will be there forever. Same energy, doesn't matter what age, I will be good. Because we need to see the change every time. That means that we have to work hard. So thank you so much for everyone. I think it's a great feeling for me coming here and spending some time with my friend and also all the students. And thank you so much.
Thank you, sir. Now we would like to take a moment to honor the hard work and the dedication of every participants. Now we will begin the distributions of certificate to all those who have successfully completed the workshop. I request Dr. Anita Nagaraju, Department of Biochemistry, Mangalore University, to read out the names of the participants.
I congratulate all the participants for successfully completing this program. And now I request Dr. Nagaraj Nagatihalli to do the kindly present the participation certificates to participants. Now I read out the names. Professor G.J. Satisha, Department of Biochemistry, Kuvempu University. Mr. Sai Kumar, be ready. Sai Kumar from CSIR, CFTR and Mysore. Miss Sadhana. Sadhana, Davangere University. Miss Aisha Siddiq, Kuvempu University. Mrs. Sneha Mohanan. Sneha Mohanan. Okay. Miss Bhavna, Mangalore University. Bhavna. Umesha Kavindi. Umesha, Mangalore University. Umesha Kavindi, Mangalore University. Sinchana, Mangalore University. Thank you, sir. Now I request Professor Manjaya, Dean Faculty of Sciences, to kindly do the honor. Smriti Kaverama, Mangalore University. Miss Sandhya, Mangalore University. Miss Aisha Fatima, Mangalore University. Mr. Gagan, Mangalore University. Mr. Harisha, CR, Davangere University. Miss Lynn Sabrina. Now I request Ganesh Sanjeev sir to do the honor. Miss Lynn Sabrina. Professor Shivasharana CT. Okay, sorry. Mr. Vijay Kumar. Mr. Vijay Kumar, Karnataka University, Dharwad. Dr. Sujata Hanumegowda, Bangalore University. Ms. Shruti, Mangalore University. Mr. Mohammad Taha, Mangalore University. Ms. Swati, Mangalore University. Last. ಅಲ್ಲ, ಇನ್ನೂರು ಜನ ಇದ್ದಾರೆ. Mrs. Neha, JSS AHIR. Dr. Raghu, Kumimpu University. Ahmad Jafar, Mangalore University. Thank you, sir. Thank you, everyone.
Thank you, ma'am. We are privileged to have Professor Manjaya, Dean Faculty of Science, with us today. I invite him to deliver the presidential address. Over to you, sir.
Namaste, very good afternoon to all. Respected icon of this GIAN program valedictory, none other than Professor Nagaraj Nagathelli. Respected coordinator, Professor Chandrasekhar Joshi ji, Professor Ganesh Sanjeev ji, local coordinator. Respected my dear colleagues, professors, various other faculties of the different locations, my dear creamy students and research scholars. Indeed privilege stand before you, such a wonderful global initiative academic network that gives some strength to research provoking thoughts. Probably our own culture, known personality within the four wall, giving such a wonderful provoking thoughts. You have to be remember ever, because in which way two of the professor given feedback, distance is not a matter, time is very much precious. Professor Nagaraj Nagathelli spent a valuable time on behalf of the department, on behalf of the university. Kindly I request all of you, give a round of applause. Sir given only on the soft core related, you have to go through only through the system, desktop and the presentation, whatever it may be. We have a real focus because these are the glimpse of us, how we can take into reality. Not only just for the knowledge based insights, but those who are not in a position to coming forward for the publications and which related to the funding, grants for funding writing, but these are all some more initiation. Why can't myself? Because sir given very clearly what, where, why, correct? No, these are all the observation, big question mark. Our own personality bringing such a way that why can't myself stand and make a strength to the society. Obviously, you have to be open our eyes, food for thought. Whatever the sir is given, 5 days provoking thoughts, which are only the small level of, glimpse level of ideas. But today, because especially this belt, sir, 7 medical colleges in this place, Dakshina Kannada. Because what you specified, your area of exposure like a cancer, you can get a real samples here directly. Probably those who are very much interested, we can make sure such a way that wonderful of our research related cancer observation, you can take a real-time applications, and today like a technological approaches in which ways are given different levels of the reading lines, which are highly motivated to us. Once again, on behalf of the university, I am very thankful to you, Professor, because we need your kind support and the good academic and research relationship because of none other than Professor Chandrasekhar Joshi. We are bringing you here and still we expecting you much more. Not only the GIAN and different level of a scheme which are going to be ventured through the Department of Science and Technology and BD, different project scheme also, expecting your positive support in all respect for the growth of the university. And especially for the participants, please make sure yourself, these are only within the 4 walls, and see the outside environment in which we can enhance our knowledge based environment, because sir in which way given all sort of the presentation and where you have to be start, where you have to be go with the different type of phases of the processing, and finally these are looks very good productivity. Then only whatever the time you spent here, valuable time is spent here, which are going to be real focusing and real understanding, and finally is going to be successful. On behalf of the university and on behalf of this GIAN program, personally, once again, I thank you each one of you for the successful of this endure level of this GIAN program, and very thankful to the Professor Joshi sir, because in which we initiated and we are doing, because we are trying our level best in which we can find out to expand our relationship with the university and the other university also. That is a GIAN, not only the knowledge base with respect within the 4 walls, we can find the reality of the good relation with the different faculties also, that motivates us and that automatically stand before us to do well because of this GIAN program. Once again, thank you and best wishes for your plan of action through this GIAN program, each and one of you present here. Once again, and thank you very much to Professor Chandrasekhar Joshi. Once again, thank you one and all. Have a good day.
Thank you, sir. Before we conclude, we must take a moment to express our heartfelt gratitude to everyone who made this event possible. To officially extend our thanks, now I request Professor Chandrasekhar Ji Joshi, Coordinator Biochemistry Program and Coordinator GIAN Program, to deliver the vote of thanks.
Good afternoon, all. So it is an honor, privilege, and to stand before you today and extend vote of thanks in this valedictory function. First and foremost, when I approached Dr. Nagaraj, usually we call him as Nag or Dr. Nag. We discussed a lot about this program, and he was very much interested, and he was ready to guide the young minds, and he wanted his knowledge to be shared, and that's what he usually used to discuss with me, what best way we can implement this kind of program. And thankfully we got a program like GIAN. Professor Ganesh Sanjeev, he suggested about this program, and we could conduct this program. So for coming over here as a resource person and sharing the thoughts of his research as well as his views, and what best way the research can be done, whatever the other plans he was discussing with the students and igniting their minds with his thoughts. So I, on the behalf of GIAN program and Department of Biochemistry and Mangalore University, I would like to thank Dr. Nagaraj Nagatihalli for being here and shared valuable insights with the participants. Thank you, sir. A heartfelt thanks to Professor Manjaya, Chairman, Department of Computer Science, Dean, Faculty of Science, for delivering the presidential address. Thank you, sir. So I also extend my appreciation to Professor Ganesh Sanjeev, because he was the key person actually making this one happening, and for his dedication and invaluable contribution in planning and execution of this event, I thank Professor Ganesh Sanjeev. Thank you, sir. So I also thank our Honorable Vice Chancellor as well as Professor Dharma, as well as Registrar Sri K. Raju Mugavira, Professor Devendrappa, Registrar Evaluation, Professor Sangappa, Finance Officer. They were actually motivating and helping us to conduct this event in a smooth fashion. I also thank National Coordinator GIAN IIT Hyderabad and the Ministry of Education for their invaluable support in the funding and facilitating this program. Their contributions have instrumental in ensuring the smooth execution of this initiative. So I extend my sincerest thanks to the directors of various centers, heads of the departments, and senior faculty members, and all the teaching and non-teaching staffs and students, and along with the center of attraction, the participants of this program, for witnessing this kind of events. Thank you all. A thanks to Ms. Rajay also, because he was instrumental in maybe telecasting this program in a nice manner, keeping all the cameras, and in a right fashion he was carrying out all these things. Thank you, Ms. Rajay. I also thank engineering section and computer section for facilitating the live telecast of this event. I also extend my gratitude to the Chandrasekhar photographer, and now a young man, he is taking the photograph of us and helping us. Thank you very much. A special word of appreciation to my colleagues, research scholars, and our dedicated students for their hard work and commitment in organizing this program. Your teamwork and dedications have been commendable. I would also like to recognize non-teaching staff whose behind the scenes efforts have played a crucial role in success of this event. A special thanks to today's Master of Ceremony, Miss Nikita, as well as the prayer that was sung in the inaugural function. It was actually lauded by even our Honorable Vice Chancellor, Ms. Shravya Nayak Group. I thank them a lot for their wonderful efforts. Finally, my deepest appreciation to each and everyone. One of you who has maybe supported us directly or indirectly in making the inaugural functions of the GIAN program a grand success. So I thank each and everyone directly or indirectly who has supported this program. Thank you one and all. Thank you.
Ladies and gentlemen, as we come to the end of this valedictory ceremony, I would like to take a moment to thank each one of you for your presence and participation throughout this workshop. It has been an enriching experience, and we are grateful for the knowledge shared and the enthusiasm shown by you all. We hope you leave here with the valuable insights and inspiration for your future endeavors. I wish you all the best in your continued learning journey.
Sorry for interrupting. I forgot to mention Senior Professor Satish GJ is here from Coimbatore University witnessing this program. I would like to thank him also, as well as Dr. Raghu. He is another faculty from Coimbatore University. Thank you, sir. Sorry.
Once again, a heartfelt thanks to our resource person and all the dignitaries, senior faculties, teaching and non-teaching staffs, and all my dear friends who are present here. Thank you all once again for your presence, and I wish you a wonderful day ahead. This is Nikita Kikan from second year biochemistry program, signing off as the Master of Ceremony. Now I request everyone to rise for the national anthem. I invite Shravya